experts from home and abroad to enjoy the 2024 Global Rare Disease Research Symposium and the second China Rare Disease Research and Translational Medical Research Annual Conference. First, on behalf of the National Clinical Laboratory Center, to express our sincere congratulations to the convening of this meeting. The rare disease has a lower incidence with multiple diseases. It's quite complicated, but the total population is not small. According to the statistics, there are about 7,000 kinds of rare diseases. The criteria or the standards for rare diseases in different countries are different according to China's Health Commission's rare disease list. In total, we have 207 rare diseases. We have about 20 million rare disease population. They are facing the difficult diagnosis, difficult treatment. The laboratory test results as the evidence for the rare diseases and the major evaluation for the rare diseases. Its accuracy and reliability is very important. The National Laboratory Center, as the national level quality control and the technique support organization, we have been long committed into the genetic disease screening and the standardization of the laboratory work by developing quality standards and the industrial SOP, and by organizing the inter-laboratory comparison, we could, regarding the genetic testing and diagnosis control, we provide technical support. Regarding the genetic screening and the laboratory quality control, we are playing a unique role. Our center has started the quality evaluation plan for 20 rare disease examination, which established the largest newborn genetic and metabolic database. We could guarantee the accuracy and reliability. In recent years, Chinese government has been increasing the investment into the rare disease prevention and guarantee. We have been improving the capability of diagnosing and treating rare disease. However, its incidence is really low and with multiple types. It is still a challenging condition. We need to develop the multidisciplinary collaboration. We need to promote the omics, big data, and AI to use them in the screening and the diagnosing of the rare diseases. We are supposed to introduce and develop advanced testing technologies to complete the China Rare Disease Database to provide scientific and comprehensive health management plans for patients. The convening of this symposium is to promote the academic communication and collaborations in the rare disease, to promote the treatment of the rare diseases to better serve patients. National Clinical Laboratory Center will be actively engaged in all kinds of researches in rare disease industry to continuously expand the types of the rare disease detection and diagnosis, to making creative quality control and laboratory tests to continuously to help to improve the capability of diagnosing and treating rare diseases to make our due contribution to the rare disease industry. Finally, I wish a complete success of this symposium. I wish you all expert scholars during this exchange, you may have more wisdom ideas. Let's work together to make our own contribution to improve the diagnosis and treatment level of the rare disease in China. Thank you all.
For remarks. 感谢马教授的精彩演讲。
Thank you, Director Ma, for your excellent remark. So today's symposium, we have gathered all the rare disease scholars and experts from all over the world. So this would be a great exchanges. So in order to memorize this precious moment, so guests who are sitting in the front, please come to the stage to take a group photo.
Row to come onto the stage for a group photo. Welcome to come onto the stage. We know that the 2024 Global Rare Diseases Research Symposium brings together hundreds of guests, speakers from global universities, hospitals, academic institutions, and pharmaceutical companies. And over the next 2 to 3 days, the guests will discuss a wide range of topics, including basic research on rare diseases, gene and cell therapies, investigator-initiated trials and clinical studies, drug development, and international research collaboration. So, I think it's gonna be a feast, right? So everybody is looking forward to the discussions and conferences in the next couple of days. 可以说，这真的是一个非常值得。
This is a memorable moment. So, photographer, please, to take this moment.
To mark this unforgettable occasion. The conference is expected to last 2 to 3 days, and during the conferences, there will be many panel discussions where guests from all over the world are going to discuss a wide range of topics, including basic research on rare diseases, gene and cell therapies, and so on. Let's give them a big round of applause and welcome them once again. Thanks for your participation. 我们再次热烈的掌声，感谢各位领导和嘉宾的到来。
Thank you again.
感谢各位。Let's go back to your seat. Thank you. Thank you. Really appreciate your time. 接下来我们还将会有更加精彩的。
Next, there will be more excellent sessions. So please be seated soon.
Thanks for your cooperation. 在合影环节之后呢，我们即将进入呢，就是我们。
After the group photo, we will start the keynote speech.
Big round of applause and welcome Professor Harvey F. Lodish, the founding member of the Whitehead Institute, Massachusetts Institute of Technology, and the member of National Academy of Science of the United States, and the fellow of American Academy of Arts and Science. And he's going to deliver a keynote speech to everyone here. And the topic of his speech, as you can see here on the screen, is the new technologies and building successful biotechnology companies to treat rare diseases. Let's give them a big round of applause and welcome him. Now the floor is yours.
This.
Thank you for that generous introduction. It's a pleasure and an honor to be here. We're all together to understand how best to treat and diagnose rare diseases. A major point I want to leave you with is that to accomplish these feats will require establishment of for-profit companies that can carry out, on the one hand, the DNA sequencing and diagnosis of the thousands of rare diseases that affect millions of patients. And I'll begin with a brief discussion of the importance of diagnosis of the diseases, particularly to advise families about genetic disorders and prenatal counseling. I'll talk about Genzyme, a company that I helped found about 40 years ago that developed the first therapies for rare diseases, enzyme replacements. And then a more general discussion about my own experiences in starting and building biomedical companies primarily focused on therapies, emerging therapies like gene therapies for rare diseases. And finally, I'll discuss a class of rare diseases, haploinsufficient genetic disorders, that really expand our concept of what a rare disease is. These are a class of several hundred genetic disorders caused by loss of function of only one copy of the gene. I'll describe some of these diseases, particularly Dravet, a devastating childhood epilepsy, and how one of my companies, Tevard, is developing very different kinds of gene therapies that we believe can be used to treat these diseases. Now, it's not working. Or maybe I don't press it. This.
Yes.
There it goes. Okay. I'm a basic life scientist. I've been a professor of biology and biological engineering at MIT for over 50 years. But I've also been very much involved in the biotechnology industry. I worked with the Commonwealth of Massachusetts to establish and administer a state fund of $100 million per year to develop biotechnology in Massachusetts. Monies that went for infrastructure, building facilities that expanded our abilities to develop therapies for many diseases, internship programs to support young people to work as interns in biotech companies, to understand how important the work was and to create career opportunities for them. I've also been privileged to serve on the Board of Trustees of Boston Children's Hospital, one of the great research pediatric hospitals in the world. And I work with the leadership of the hospital to educate the research faculty at the hospital to become entrepreneurial and to understand how to take research from their laboratories into the broader communities by establishing biotech companies and by working as advisors to these companies. Many advances have emerged from this, including one of the very recent gene therapies that have been approved for sickle cell anemia. I've been involved in starting over a dozen biotech companies. Many of them in red focus on rare diseases. So I've had the privilege of working with many groups on diseases and helping them think through how to take the advances in our laboratories into the clinic. I need to begin with this federal law, the Orphan Drug Act of 1983, which promoted in the United States the development of therapies for rare disease, giving market exclusivity and certain financial advantages to companies developing therapies for fewer than 200,000 American citizens. This law was essential to develop Genzyme into a company that developed therapies for rare disorders. And I would encourage China and many other countries to pass similar laws to enhance development of rare disease therapies. Just to make the obvious point that I'm sure many of you know, that many genetic rare diseases are autosomal recessive because of consanguineous marriages that occur in many, many communities throughout the world. I have here just two examples from fairly small European countries, Iceland and Finland, of rare genetic disorders that are almost exclusively found in these countries. Pakistan has a high level of consanguineous marriages. I am told 1 in 1,300 children has a rare genetic disease. Very few of these have been understood and have been mapped genetically. A point that we often don't think about of rare diseases, because many are autosomal recessive and are confined to specific ethnic groups, prenatal counseling, such as this program in the Orthodox Jewish communities throughout the United States, this prenatal screening done not by medical officials, but by the religious leaders of the community, the rabbis, have counseled young people and have almost eliminated many of these horrible genetic diseases like Tay-Sachs in one generation. So let me tell you a bit about building Genzyme, the first company to develop therapies for rare diseases, a bit about my own experiences in starting and building successful companies, particularly those that focus on rare diseases. And then finally introduce many of you to haploinsufficient genetic disorders and the challenges in developing treatments for them. I was one of the founders of Genzyme in 1979 with 7 colleagues from MIT in the science and engineering faculty. We were all leaders in our respective scientific fields, but it was this gentleman, Henry Termeer, whom we recruited as CEO of the company. It was Henry, not us, who had the business background to realize that one could start and build a company making therapeutics for rare diseases. That was his vision based on his experience in a number of biopharmaceutical companies. And we decided that the first therapy we would make would be an enzyme replacement for an autosomal recessive lysosome storage disease called type 1 Gaucher. It's a disease in which a particular lysosomal enzyme, glucocerebrosidase, is missing. In the most common form of the disease, type 1, the central nervous system is spared, and the cells that are affected are primarily macrophages throughout the body. And as I'll describe, we developed an enzyme replacement therapy to target the enzyme to macrophages where it was taken up and reverse the cause of the disease. Gaucher, like many genetic disorders, autosomal recessive genetic disorders, is enriched in particular ethnic groups, in this case, descendants of Eastern European Jews. We targeted the protein to macrophages where it was taken up by receptor-mediated endocytosis and through the normal endocytic pathway was trafficked to lysosomes. To accomplish this, we took advantage of a receptor on the macrophage surface, a protein that binds polymers of mannose. This mannose receptor originated or evolved as part of the innate immune system because many pathogens such as fungi, other yeasts, have polymers of mannose that coat their surface. And here you can see a macrophage enveloping a yeast particle based on the binding of the cell surface mannose receptors to the polymers of mannose on the yeast cell surface. To apply this to Gaucher, we made the recombinant protein, glucocerebrosidase, in Chinese hamster ovary cells in culture. The protein was secreted with a common complex asparagine-linked oligosaccharide, where it was terminated by sialic acid, galactose, and acetylglucosamine, the sugars colored in yellow. And we removed these outer sugars by treating the recombinant protein with the 3 enzymes that are indicated on the figure, converting the oligosaccharides attached to glucocerebrosidase to a 3-mannose core, which is common to all asparagine-linked sugars. This was sufficient to target the recombinant protein to macrophages. The enzyme replacement is given by infusion. Here you see a young boy with Gaucher disease, greatly extended liver and spleen. He's also quite anemic. Within weeks of treatment, he became completely normal. This is one of the reasons I wanted to explain to you how we developed the Gaucher drug. The second reason is very personal. As I mentioned, Gaucher disease is most common in descendants of Eastern European Jews. I left Genzyme at the initial public stock offering in 1990. 12 years later, our oldest daughter was pregnant. She and her husband are of Eastern European Jewish descent. They were given a battery of prenatal tests, and it transpired that both of them are carriers of type 1 of Gaucher disease, which meant that the child had a 1 in 4 chance of having Gaucher. Now, I had no idea when I helped start Genzyme that Gaucher disease was in my family. Our daughter had no idea that her father knew anything about this, had amniocentesis done, but had me take the call from the genetic counselor. Dr. Lotish, I'm really sorry. The child your daughter is carrying has Gaucher. I asked for the alleles because I knew if there were no enzyme, that it would be a neurotropic disease leading to early death. But I could conclude from the alleles that she told me that it would be type 1 Gaucher. This was, of course, later confirmed by the genetic experts at Boston Children's Hospital. I'm pleased to report that Andrew is now 21 years old. Here you see him beginning his infusions with the enzyme replacement therapy that his grandfather helped develop. Incredibly, he has never been sick a day in his life from Gaucher disease. He is a pre-medical student at Wesleyan University. He's an athlete. He rode across the United States. And this is why we are all in the business of trying to treat rare diseases. You never know who will be helped by your own research. Genzyme, of course, went on to develop enzyme replacement therapies and other therapies for a large number of inherited genetic disorders. Very briefly, I want to describe to you my own experiences and understandings of starting biotechs. Because as with Genzyme, it would have been impossible to develop a therapy for Gaucher disease without having a for-profit company. And what I trust you all understand is that the research we hear about in our universities, in our research hospitals, they do not develop drugs. At MIT, we do not develop drugs. We develop what is called proof of concept. That is a molecule that might work in a cell culture system, or something that works in a mouse or another animal to treat a disease, or even in a few humans. But to actually scale up the manufacture, being able to make the material reproducibly under good manufacturing process, to get all the regulatory approvals to market the drug and to convince physicians to use it, this all requires a for-profit company. And it requires an investment of tens to hundreds of millions of dollars because of the requirements for lengthy clinical trials. This is an amount of money that no research hospital or university can provide. So part of my mission here in Shanghai, as elsewhere I go in the world, after this I'm going to Taiwan and to Japan, with the same message. To really treat these diseases requires the incorporation of a for-profit company. Each clinical trial requires, hundreds of millions of dollars, and that's before any money is returned to the company. I don't want to go into elaborate detail, but in order to start a company for rare diseases requires an entrepreneurial faculty. That is, many of my scientific colleagues are experts in their own fields in universities and medical schools. But they also, as I do, work as advisors, as boards of directors members of companies that are developing treatments for human diseases. Besides intellectual property and a business plan, one needs a top scientific advisory board who can give the company advice from the leaders in the field how best to develop drugs for a particular indication. And as I said, it requires financial backing and, importantly, supportive infrastructure by governments and regulatory leaders. At MIT, faculty, are given 1 day a week outside consulting privileges, where we can work, as I do, for not-for-profit corporations and institutions, but we can also serve on advisory boards of directors of for-profit companies. And I argue that the success of biotechnology companies in the United States, particularly in the Boston area, has its root in this policy of encouraging the academic leaders in so many fields to start and work with and consult for biotech companies. Again, I won't burden you with it, but one needs a business plan. One also needs trained workers in companies. That is, to be a successful Genzyme, you need, yes, PhDs and MDs who guide the company's fortunes and research at a very high level. But the vast majority of the people working in biotech companies are lab technicians, marketing experts, financial planners, and so forth. Do not need necessarily advanced degrees, but need training. So as you plan to develop biotech companies here in China, Shanghai in particular, think not just of the leadership of the companies, but of all kinds of employees that you need to build companies for rare diseases and other things. You need boards of directors, scientific advisory boards that I've already mentioned. Let me finish briefly by introducing this class of haploinsufficient genetic disorders. And I'll describe one of my companies that is developing therapies for them. The rare diseases that we are most familiar with are autosomal recessive, autosomal dominant, X-linked, but essentially genetic disorders that one inherits from one's parents. These diseases are different. They are caused by mutation very early in the child's development. It's not known exactly where, but most likely, that was a 5-minute warning, most likely in the sperm or the egg, where only one copy of the gene is inactivated. As I'll elaborate, many of these genes are highly regulated, expressed at very different but important levels in different cell types. Many of them affect development, particularly of the central nervous system. And they're very difficult to diagnose, and almost all of them require either whole exome or whole genome sequencing. The disease that I became familiar with, it has not my own laboratory work, but through two gentlemen, Warren Lammert and Daniel Fisher, have daughters with Dravet syndrome. Again, it's a de novo mutation in a gene termed SCN1A that encodes a voltage-gated sodium channel, NAV 1.1, that is expressed throughout the brain, but at high levels in inhibitory GABAergic neurons. And the disease is characterized by extensive epileptic seizures that can be managed by treatment with drugs. But these drugs do not affect the overall developmental course of the child. Defects in normal development, many of the children are unable to speak and communicate, many other problems besides epilepsy. These mutations occur at many places in the voltage-gated sodium channel gene. And the gene is characterized, we call it a Goldilocks gene, where too little is bad, but also too much also causes a disease. So the issue is restoring the normal level of the protein in the normal level of whatever cell it's normally expressed in. I'll skip over these other genetic disorders, but to point out one way of doing it is to lengthen the stability of the wild-type copy of the message. Human messenger RNAs are in general short-lived, with an average half-life of 3 hours. That means if we can lengthen the half-life of the good copy of the SCN1A message by as little as an hour or two, we can greatly increase the protein output over its lifetime and thus reverse the effect of the loss of the other allele. One way of doing it is based on a technology developed by Dr. Jeff Collar, who is, again, a leading scientist, but also on our scientific advisory board, and we consider him a co-founder of the company. What he does is take advantage, simply put, that poly-A sequences are at the 3' end of normal messenger RNAs, and because of a protein that binds to these sequences, the poly-A binding protein, mammalian human messenger RNAs are stabilized. And what he did very cleverly is take a CRISPR protein, Cas13, that targets RNA sequences, guided by a guide RNA, a CRISPR guide RNA. It will bind to the complementary sequence in an RNA. Normally, it will cleave that sequence, but he inactivated by mutation the nuclease site and linked it to a piece of the poly-A binding protein that from his work we knew would stabilize messenger RNAs. And this chimeric protein that you can see here on the left, the CRISPR Cas13 binds it to a target sequence in the messenger RNA. The appended poly-A sequence stabilizes the messenger RNA. And very simply put, you can see on the left, combining the CRISPR and the poly-A binding protein, we see an increase in the target messenger RNA. This is the mRNA for Rett syndrome. One more minute. On the right, one can see that by positioning the target RNA at different positions in the 3' untranslated region, one can upregulate the messenger RNA and make more protein at different levels. Here we see it with another genetic interesting gene, SYNGAP1, where haploinsufficient mutations also cause epilepsy. There are other protocols that other companies are developing that will also, by different mechanisms, upregulate the wild-type copy of the messenger RNA and are also being tested for a diversity of haploinsufficient genetic disorders. So to leave you with, companies are very important in developing the therapies for the rare diseases that we all want. Haploinsufficient genetic diseases, like many others, can now be tackled by clever scientists working in biotech companies. Thank you very much.
Thank you very much. Thank you very much for your wonderful speech. 那么接下来的重磅演讲嘉宾
Next, our speaker would be the academician of the Chinese Academy of Science and the president of Fudan University, and academician Jin Li. Because of some important business, he could not come here, so he has pre-recorded videos. So, dear experts, good afternoon. First of all, on behalf of the hosts, Fudan University, to re-welcome all of you to come to the 2024 Global Rare Disease Research Symposium and the second China Rare Disease Research and Translational Medical Annual Conference. Due to some urgent business, really my apologies. I have only to communicate with you with the online video. So my topic today would be from genome to phenome. Actually, we all know phenotype is the human features or the measurable human features, and phenotype phenotype is determined by gene and environment. As for phenome, refers to all physical, chemical, and biological combination from the birth to death of an individual. Well, the core of the biological research is to figure out the relation between the macro phenotype and the micro phenotype. Macro phenotype means what we could see with naked eyes, the structural and behavioral phenotype. The micro phenotype refers to at the molecular level some features. In the past, they were not measurable. Nowadays, they are measurable only by building the relationship between the macro phenotype and the micro phenotype. Then could we study and understand the mechanism for the macro phenotype. In the last 4 decades, in the biomedical industry, the momentum was from the Human Genome Plan. Whereas after the Human Genome Plan was completed, we found a large number of disease the phenotype, the relation between the phenotype and the sites of the genetics, and we reviewed some new biological process. However, we also see the human genome project, although the sequencing is easy and relatively is cheap. However, nowadays the new ideas, there are less and less new ideas that could be reviewed. Where is the bottleneck? I think to a large extent it is due to the complexity of the genotype. In the past, we call him the genetics in the genome era, which means to figure out the phenotype first, especially the macrophenotype, the relation between the macrophenotype and the gene, then study its molecular phenotype with sequencing. However, the genome, especially after completion of the Human Genome Project, it provides us a lot of new findings, and it promotes the development of the precision medicine. However, the genome research there encounters a bottleneck due to various reasons, due to the complexity of genetic mechanism, complex influence on disease and health of environmental exposure, and complexity of the phenotype, which means on the one hand is gene, on the other hand is phenotype. If you really want to figure out the linkage, there are so many links requiring step-by-step connection, and the relation is not like a causal relationship. So the previous study methodology encountered a lot of difficulties. Well, one way is to reduce its complexity, like the whole genome sequencing, and to study the interaction of genes to increase the sample size from hundreds individuals to 10,000 samples nowadays to millions of samples, and also the epigenetic effects. Well, another one could be, is there a way to reduce the complexity of the genotype, the phenotype? So here we talk about the perspective of the phenome, which means on top of the previous methods, trying to link the relation between the macrophenotype and the microphenotype. In the past, we say, since there are multi-omics methodology and multiple phenotype measurement methods, could we measure all phenotype to figure out the relation between phenotypes? then to see what role does the gene play in this process. The purpose is to reduce the complexity of the phenotype. In order to do that, maybe, and maybe, just like the genome project plan, by studying the relation between the phenotype, it could promote the biomedicine development. Say, for the macrophenotype and the microphenotype, if we could further link them, and for many underlying molecular mechanism of the macrophenotypes, we could achieve more precise disease classification. In order to do that, we have been promoting the International Human Phenome Project. Why it must be an International Human Phenome Project? Well, of course, among Chinese population we could do it, or in a certain ethnic group or in a certain nation we could do it. But the issue is considering the phenotype itself in different ethnic groups, in different nations. Actually, there are significant difference. If we do not make good use of such difference to increase the abundance of the phenotype, it would be a huge pity. But here, in order to do it on a global level, then we are encountering a huge challenge. So the data generated from different labs, how to standardize. Therefore, we have to standardize the technical requirements. That is the first step. Meanwhile, the data generated in different labs must be shared. Otherwise, the data generated, its role could be very limited. So we hope to see the multiple labs in the world to generate high quality data, and they are comparable data. So, with this big scientific target, first we want to figure out the relation between phenotypes. We call it the Human Genome Atlas. Why we call it an atlas? Because if you want to connect the different genotypes of the human beings during the research, actually usually we could start from one point with the connection point, then we could find out the future new direction. So this is like a navigation for the study direction. While promoting this International Human Phenome Conference in 2015, we convened the Shanghai meeting, and it was officially launched in 2018. Although we had COVID-19 pandemic, but year by year, we had annual meeting, and with those annual meeting, we had reached a series of consensus. So we formed this International Human Phenome Consortium, nowadays covering scientists from 20 countries. Like we just mentioned, in the last 4 years, we reached 4 consensus, including what does the phenome do, and how to share and develop data, and SOPs regarding the measurement standards and the standards of the procedure, how to standardize them. Additionally, the phenome systematic study requires a data platform. We call it PhenoBank, which will be online this year. The issue would be, in order to measure a lot of phenotypes, what to measure? Could we measure them? So at the very beginning, we want to try to measure more. So supported by the Shanghai city, in Shanghai, we built a experiment platform which could enable us for molecular phenotyping, cellular phenotyping, imaging phenotyping, and biological phenotyping. So we could measure more than 24,000 phenotypes for one person. In order to have good quality control, we need standard material. Therefore, we developed 8 first standard reference material, where this is still ongoing. So those standard reference material could be used for the DNA analysis, metabolic analysis. So this is the national level 1 standard reference materials. and it has been recognized by the EU. At the very beginning, in the last 5 years, we measured 1,000 individuals with more than 24,000 phenotypes in each individual, and we came up with our design for the framework of the phenotype. Based on those 24,000 phenotypes, we established a framework as an atlas. We call it Human Phenome Atlas 1.0. Left one. This slides you see, there is a cartoon. That was the atlas. Each dot represents a phenotype. Between dot and dot, if there is a strong relation, then a line will link them.
Based on our initial analysis for the 24,000 phenotypes, we found 1.5 million strong associations, and they are of great value for further studies. Based on these leads for further studies, we've had initial analysis, and we proposed high-value scientific questions. Through such analysis, it is a very important way of finding new questions. Through the associations of phenotypes, we propose new questions for study. It's not providing a solution, but those questions will bring us to more mechanisms for biopharmas, and it could be new targets for therapies, and we can make new biomarkers to block those processes. Let me give you some examples. We've got some interesting results. Case number 1. And we've found limb development genes influencing the formation of fingerprints. We have 10 fingers, and in the upper part we have fingerprint patterns. Genetic experts have been doing research about the genes determining such patterns. There were no breakthroughs. You see, the inheritance from parents to children is very complicated, and we've used ways of thinking originating from phenome, and we've found that there are genome-wide associations, which are 43 of them. And these 43 genes are related to the development of limbs rather than skins. One of the key gene is EVI1, and we put that EVI1 and we knocked it into mice, and we see there are some changes in the patterns of those mice. And we find new molecular biomarkers, and there are combination of 5 serum metabolic molecular phenotypes, and they could predict the incidence of Alzheimer's disease in the coming years. And also in terms of cancer studies, we found the biomarkers of DNA, and 4 years prior to the cancer, we can identify possible GI cancers. Furthermore, for people in plateaus who go to altitudes, there are different reactions. And what is the mechanism at play? And we found new mechanisms, and those new mechanisms could be targetable, and we design drugs targeting these, and we have made MT8. R&D work is almost done for this. For the aging mechanism, what is essence aging? And for a cohort of 1,000, we've had phenomic studies. In the aging process, we look at the network structure and the specific features, and we shed some light on some interesting mechanisms. The theme of this conference is for rare diseases, and we have established a library and AI. Little Boo is a doctor robot, and this is used in the hospitals. For the autism spectrum disorder, we have used phenomics, and this is providing a new paradigm. And through phenomics, we screened new biomarkers, and by using iPSC-derived disease model, we have had validations that provide further support for modeling. For clinical immunology team, they've tracked their cohorts with immunodeficiencies, and you can see REPK1 that is activated, and they look at the MOAs for the regulations. And our GI team, following the children cohort of IBD, studied SYK that lead to IBD, and animal models were tested, and we proved that the SYK inhibitor and bone marrow transplantation can be of value. And for interleukin-10 related genes, we have studied the pathogenesis of IBD. And for the treatment of beta thalassemia, there are new strategies, and we have studied investigator-initiated studies. Then our dermatology team had studies, and they have found 6 genes leading to skin disease, and this is COAP and DIDA. Looking forward, we believe phenomics is at a critical juncture. This provides clues for further studies in the future, but we'll come to a certain specific disease in one organ, in the cells, in the molecular mechanisms. We need to further expand the new modalities. One of them is molecular imaging. With molecular imaging, the new biomarkers, new mechanisms can be shown in the imaging, and we can look at its distribution in the whole body and look at the functions possibly. To carry out research on a cellular level, the organs, tissues, molecules should be connected, and now we have special omics, and this is connecting the macro and micro phenotypes, and this is the last mile. By doing research in spatial omics, we have made plans, and this is going on. Hopefully, in the measurement of those dimensions, we hope we have a controllable environment. This is not only for measurement. You see, for phenotypes, it is interfered by environmental factors. Our way of thinking is to make sure we have a simulation cabin. All tests can be carried out in the simulation cabin, and the environment can be precisely controlled. So we proposed to build the system biology research facility. This is for real environment simulation, then multi-scale phenotyping, and this is now with a good foundation for the future measurement and analysis we can integrate the work. With that, I thank you very much. Thank you for your listening. And correct me if I'm wrong. Thank you very much.
好的，再次感谢金院士。
Thank you, Professor Jin, for your wonderful presentation. Now, I'd like to invite co-founder and Secretary General of Hope for Rare Foundation and the founder of CORD, Mr. Kevin Huang, to give us a presentation. Okay. Thank you very much. It's great to be here, to have this opportunity to share something with you. I will talk about the Hope for Real Foundation. In China, we'll promote scientific researches, and taking this opportunity, I'd like to brief you about the concepts, philosophies, and report to you about the progress we made. I'm sorry, those two screens are different. What I see and what you see are different. When you look at the slide on this screen, you see our foundation is promoting research and translation medicine. We'd like to be an engine for innovation. This is the role of this foundation, and this is our mission. In this global symposium we have in the 3 days, this is what the foundation have been doing concretely with specific steps. I'd like to report to you what we do and in promoting research and translational medicine for rare diseases. I have my idea. I'm not a scientist, I'm a patient with a rare disease. Standing here, I don't have confidence to talk about science. But in the last decade or so, I've been promoting the advocacy to patients, services to patients, and development of industry. In the face of the scientists, as a layman, as practitioner in this industry, I share some of my ideas. I hope you could have discussions with me. From this slide, you could see these are famous celebrities who are familiar to us. For these celebrities, they have or had rare conditions. Let me tell you this. Rare diseases are not so far away. Take me, for example. My parents are normal persons. My whole family do not have any person who have a similar disease like mine. How come that I have it? So, I pose this question to you. You know, every person could be related to a rare disease. As Professor Harvey Lodish shared his experience to develop Cerezyme to treat Gaucher disease type 1, and by coincidence, his grandson had this condition. See, rare conditions are related to everybody as long as there is life. There are possibilities of having a rare condition. So, let me share this with you. For the scientific research regarding rare diseases, are we doing enough? I have cruel facts to share with you. There are over 7,000 known rare diseases in humans. Actually, it's more than 7,000. It's a huge basket with so many different conditions. However, only around 700 drugs, therapies have been approved for rare conditions in the world. This is only 6%. That means 94% of the rare diseases cannot be addressed with an effective therapy. Hereby, I salute the scientists. Please keep your hard work. Find more therapies for the patients. Among the 94% of rare diseases without treatment drugs, we need to see there are hope in the 6%. In the last 5 years, therapies have been developed, but it's not enough. There are huge challenges. Those therapies are of high prices. Globally, the most expensive therapy is 4 million USD. Think about it. For all of you, you could be rich. You may not afford such a 4 million US dollar therapy. So, in the field of rare diseases, the humankind in the world face huge challenges. However, all countries, scientists, we can go to the outer space, we can build rockets, space stations, aircraft carriers, luxurious buildings, landmark buildings. How much of the resource is spent on the unmet needs of patients with rare diseases? I would like to tell you this. In this current conference of ours, we need to reach some consensus. This consensus require us to take urgent actions. The biggest challenge for the humankind is rare conditions. We need to put our efforts to solve these problems one by one. This is point one I'd like to share with you. This calls for the global efforts of scientists, governments to reach a consensus. Number two, rare diseases are related to everybody, as life is being passed from parents to children. Your children will have children of theirs. Nobody is exempt from any condition. Next consensus, thirdly, hopefully, we could realize why there are unmet needs for 94% of the diseases without any therapy. Because for rare diseases, market is not taking its role. You cannot rely on the market dynamics to solve the problems. For example, for some diseases, there is only 100 or 500 patients who have this disease. No company would like to invest there. So for rare diseases, the only way to solve that is to pull the efforts from the public, from the policy side, from the US or EU. You could see those who are advanced in developing new therapies for rare disease have a complete set of policy framework. When market is not playing its role, we need to leverage this framework. I call for all countries and regions. Please invest more for policy frameworks. Please invest more in science of rare diseases. We cannot rely on market it only. It doesn't make sense. Number five, I hope to reach consensus with experts here today. Only through global efforts can we make a difference. It's simple. For some rare diseases, in the US, you have very limited number of patients. In some other countries, you may have only a dozen patients. How can you carry out studies in a small country with a dozen patients? For the study of rare diseases, we need to pull up those data. We need to invest in clinical studies, and we need to pull the efforts of scientists from the world. Only through international cooperation can we tackle the rare diseases. Lastly, I share this point with you. For R&D of therapies for rare diseases is for all patients. It's not only for the rich patients only. It's not only for the developed countries. They should be accessible to all. For any scientist, you may have a dream that your research results in the process of translating that you could help all patients globally. Patients from all corners of the world could have equal access to the innovative therapies. So, in the very start, I look back to the last decade. I find those points for reflection, and I share those reflections with you, and I hope we can echo in our mind. Again, I will go to this question. Have we tried our best? Have our country tried our best? Have we spent resources from the public finance, from charity to rare diseases? We may reduce investment in military spending. We may reduce spending in luxury buildings. We may reduce investment in the non-urgent matters so that we can increase investment in life sciences.
So, if this is the first part of my presentation, I'd like to share with you. I think as for the Hope for Rare Foundation, as a scientific research symposium I would like to promote in China, we want to say, China, you are coming all the way to China. This is both a stranger and also this is a familiar country to you. So what we could do? The first point is that in China, in the last 3 decades, we had rapid economy development. Now we are the second largest global economic entity. So the development of the medicine, the development of the economy, should we go after the global trend? So should we be engaged in the cutting-edge development of the rare disease technique? So as such a large economic body, we need to make our own contribution to the treatment of the global rare disease. Not like in the last 10 or 20 years, we had to license in all the innovative drugs into China. Without such medication, then Chinese patients would have no drug to use. And I still hope there are 94% of the rare disease studies. So is it possible that Chinese companies could you make like 1, 2, or 3% contribution together with the global research companies to make our own contribution, to make the Chinese contribution to the treatment of the global rare disease patients. Finally, I hope that among those contributions, we hope for a rare foundation. We could be serving as a bridge. We could serve as a promoter. So this is something we would like to do in China. From the global approved rare disease drugs, we could see among which one-third of those drugs, one-third of the early R&D of approved drugs was driven by patient organizations and foundations. I would like to ask you, investigators, in the past, you used to have some funding, scientific funding from the patient organization and foundation. Could you raise up your hands? Whoever had such fundings. So we basically see scholars overseas, they all used to have fundings from foundations. So rare disease foundation regarding the scientific research promotion, yes, it could play a big role. Although regarding the scale of the fund, we are not comparable with the national fund. However, the non-government foundation could play a unique role in the field which is not covered by the national fund. And from the patient's needs perspective, we could play a big role. We could promote the different stakeholders to do scientific research for rare disease. So that is at the very beginning of the foundation of the Hope for Rare Foundation, we studied the best 30 foundations just like us. We learned how they sponsored the scientific research in the last 2 decades. We want to start in China as well, although we are latecomers, but we'll be quick.
那我们为什么，包括我自己，我
Why,
为什么
including
会做这
myself,
件事情？
why could I do
我
this?
还是要重申，我不是一
I would
个
like to reiterate I'm not a scientific
但
researcher,
我发现我
but
这
we
个过去
found
的
that
10
in
年，
the last
我
15
做了一些
years,
对行业
I
的价
did
值的，
some
比如说，
works
我
for the
呼吁
industry
社会的
insight.
极大的关
For
注，
example,
在
I
工作中
advocate
产经
the
面
great
鲜
attention
为人
from the
知道现
society.
在的
So in the
人
past,
口
nobody knew what
那
is rare
产经面
disease.
这些人知
Nowadays,
道，我以
rare
外，
disease
成
is
为
well
了我
known.
国政府以及医
So
疗行业
besides
最重要
tumor
的一
rare
个重要
disease,
的议题。
now is becoming the most important medical topic in Chinese government. That is something I did in the last 15 years. And also,
能够发出声
I
音，在这些
enable
产
the
经面
rare
上
disease
能够做
population
到
and
我们
communities
在
in
事业
China
里面
to
让
speak
大
up,
家看到
to
我们
enter
每
our
一个产
vision,
经
to
面
let
家
us
属
see
面
each
临
and
的社
every
会
rare
的
disease
忽视，
family.
面临的
They
企业
used to
的极
be
大的
ignored
挑
by the
战，
society.
面临
They
的
were
生命
challenged by
的
the disease.
危险。
They were threatened by
这
the
是在我
disease.
过去的10年
So
的
in the
经
last
验，
15 years, that was
你在
second
过
job I
我在
did.
过去的10
For
年
my third
推
job in
动
the
了
last 15 years,
产经
I've
面
been
的政策，
promoting
让
the
中国越来
policy
越多
for
的
rare
产经面的
disease.
政策
So
能
more
够
and more
去
China's
有
policies
利
for
于
rare disease
产
could
业
be
链的
beneficial for
临
the
床的诊治、
clinical diagnosis and
诊
treatment for
断、
rare
治疗、
diseases.
康复，
Diagnosis,
能
treatment,
够对应
rehabilitation
产经面的监
could be
管，
more
能对
beneficial
应
for
产
the
经面的
monitoring
控
of
制。
the rare
这是我
disease
过
could be
去
beneficial
所带领
for the
的
payment
各个产
of
经
the
中
rare
心的
disease
各
drugs.
个公基会
So
在
in
中
the
国
past, I was leading the core team
因为我
and
过
the
去
Hope
10 年
for
经验的
Rare
深入，
Foundation. We did
我发
something
现我
because
们
of
产经面的
because
患
of
者
my efforts
仍然面临
that
无药可
our
治，
rare disease patients they were still facing
没有药
situations
我
that
们能不能在
no
国
drug
内
to use. So
和
we
其他
were
的机
considering
构一样
in China,
推动
just like in
国
China,
内
could we just like other
对
foundations
这
to
些医疗
promote
的
the
医疗的
scientific
公益
study?
基金会的创立，
Then we
我想在
founded
这
the
里
Hope
回顾
for
我们
Rare
2年前
Foundation.
在这样的
Here,
一
I would like to
个
review 2
产
years
经面
ago.
日的这个2年前的国际
2
产经
years
面日
ago,
期间，我们
during the
创立了这样
International Rare
一个
Disease
公
Day,
益基金
we
会，
created
我
this
联
Hope
合
for
了
Rare
多位的
Foundation
世界
together
的
with
科学家
multiple famous
和企
scientists
业家
and
和公
entrepreneurs
司
to
为他
support
做这
me
个事情。
to do this.
那这个公益基金会
The
就
Hope
是希
for
望能
Rare
够在未来
Foundation
推动产经面的科学
wants
行业的
to
发展，
promote the
能够
rare
去推动
disease industry
科学家
development in the
的科
future,
研的制
to
度，能够
promote
去推
the
动创发实
sponsorship
践，
of the
能够去推
scientists,
动
to promote the translational
影
medicine,
响力的扩
to
展。
promote the influential
一定要向全世
investment,
界优秀的
and
科学
also
家
just like
推
the
动
most
产经面的
the
科学研究
influential
和
global
创发
foundation,
实
we
践。
want to promote the scientific research on the rare disease and
讲了这么
translational
多，
medicine,
简单来讲，
covering so
没有什么
much.
特别要做
To
的。
be simple, what are we
我们是要
supposed
鼓
to
励
do?
科学家，
We want to
我
enable
们是要
scientists.
来资助中
We
国
want
的
to
事业的科学家，
sponsor
让他们能够
the
去
Chinese
更
and
好
the
的投入到
international
产经
scientists
面的科学研
to
究，
better
对应
engage
到
themselves in the rare
人
disease
才培
study.
养的
So
制度，
we need to
我们要去
support
搭
the
建这样
talent
的产经面
cultivation.
的
We
科
want to
研的
build
会议
the
的
rare
平
disease
台，去搭建我们的
scientific
科学
research
创新联
platform
盟，
to
去
build
支持
our
我们
scientific
的科学
innovation
家，
alliance to
去
support our
把我
scientists
们在社会上募集到的
to
资金支持我们的
to
科研项目。
give fund
每
to
一
the
个科研项
scientific
目
research.
带来了极
So
大的
each
风
and
险和
every
危险，也
scientific
会
research
对科学
program
的
is
研究
risky,
是
is of
不
great
利的，会
uncertainties.
成功的。
There's
但
no
是如
certain
果我们
things.
没有持续不
However,
断的
if
去
we
为科
do
学家
not
去投入科研的资源，去
invest, if we
相
do
信我
not
们的科学
sponsor,
家，
if we do not believe our
那
scientists,
每一个科
then
学的突破，
the
都
each
无
and
法成功。
every scientific
所以
breakthrough will
每
not
一个
happen.
科学的突
So
破、
each
科
and
学的进
every
展、科学
scientific
的发现、
development,
药物的研
discovery,
发，都是
breakthrough,
来
drug
自于
R&D
长达10
is
年、
from
20
like
年幕
10
后的
year
这
or
些科学
20
家
year
的默默的
scientists
付出。
who've been working
而我们
silently.
过
and we hope for their foundation. We are willing to support them. Finally, we hope to promote more scientists for their scientific and technological translation, which could be used in clinical practice, which is of great importance. As a patient, I know that China's scientists in the past, we published we published too many papers in the top-level journals. However, the Chinese scientists up to now, there's no almost there's no rare disease. Original rare disease drug was initiated in China and completed in China. So I want to say, to promote the translational medicine is of utmost importance. So each and every scientist, we hope you just like have a lot you could talking about the importance of the translation to translate your technique into the clinical practice. That is something we patients we would like to see. Well, of course, we've done a lot with our scientific symposium, with our scientific seminar, with our young talent sponsorship, with our scientific sponsorship and individualized exploration, and our Goldface Move Mountain project. We've been insisting, although it's difficult, but our direction is correct. So here I'd like to say for 2 minutes more, in the last 2 years, our best business is Good Faith Move Mountains. That was a patient organization scientific research sponsoring program. This is we have great characteristics. This is a new model. So we are focusing on the very rare disease. No scientist is studying or no research, no regimen for a certain disease. We want to promote the China's PI to do scientific work. However, the establishment of the scientific research program is based on clinical patients' needs. There is an old saying that the gold and stone will split if your sincerity is there. So we named our project after this one. So we want to put our clinical research into clinical use. Well, in this Goldface Moves Mountains, I would like to share our basic process. So with the establishment of this project and to complete the preclinical studies until finally we put this result into humans to solve the real clinical needs. We want to explore this paradigm with being rapid, being efficient, being low cost to promote scientific studies. This is a new model. This is with rigorous scientific research and validation. Now there are 5 projects. There are 5 ongoing projects from October 2022 to our latest project in April 2024. All those projects have been focusing on a very rare disease where no regimen is available for those diseases. We are sponsoring the Chinese investigators. Finally, I want to say, in the current society, we are moving forward in the difficulty. We are affected by the geopolitics and uncertainties. In some local regions, there are ongoing wars. In such a world, we are facing the obstacles to the development of the pharmaceutical industry. I want to say the scientific research must not stop. International collaboration shall not stop. There are more disease population. Each and every vivid life is waiting for your efforts. Is waiting the great promotion by the government and countries where they have advanced technologies to expand science and technologies to the underdeveloped regions and nations. We really want to unite the science. So I think this is a difficult task. However, it must be correct. Hope for rare. Let's together for science. So I hope the global community could be united for science and for rare disease. Thank you. So thank you, Mr. Huang, for your keynote speech. So the last keynote speech speaker would be the co-founder of the Hope for Rare Disease, and Mr. Xu Tian. Now let's welcome. I'm so happy that I could come here for this event. First of all, I would like to say to my fellow patients, families of the rare diseases, investigators, workers for the rare disease, I would like to say it's so happy to see you all. Every time I meet you, I think about you. Makes me energetic. Thank you all. Secondly, just like Ru Fang just mentioned, the study for rare disease is a challenge. But I would like to say to all young investigators, young scientists who just entered this world, the study for the rare disease is a challenge. But like all challenges we human beings are facing, challenges will activate us, will make us more wise, more smart. The challenges for rare disease study could help us to solve, to develop solutions that could help everyone. Thirdly, just like Dr. Harvey Ludish said, we need benefits to help the R&D of the rare disease drugs. So I would like to show you some cases I'm working on to tell entrepreneurs and investigators. Studying rare disease could be beneficial. First of all, thank you for your concern and support for the Hope for Rare Foundation. And also, we would like to say thank you for supporting the Westlake University for 3,000 faculty and students. Back in Yale, I studied and I did teaching and administration works for 25 years in Yale. And also next to Yale, I did some translational work study. Back in Yale, for basic studies, we were studying the methodology for genetics. We were studying the genetic signaling pathway, how to modulate the size of the organ, how to modulate the growth. Here, we had the TSC to study the rare disease, made us find the major signaling pathway for modulating the signal. First, we proved this signaling was modulating the growth. This study is not just useful for rare disease, but nowadays it has found that signaling 70% of the cancer patients is affected by this signaling pathway. Studying this signaling pathway later, due to the rapamycin, could inhibit the growth to treat the bladder cancer. However, among 40 bladder cancer patients, only one worked. Our sequencing machine was developed back then. So the surgeon, the physician, did the gene sequencing. They found, yes, there was TSC mutation for that only patient which worked. That work and the other following works for promoting and expanding the precision medicine, it worked, and we were so happy. Because of studying the TSC, my friend and I, John Rosberg, in our town, we started to build the Rare Disease Research Institute to develop diagnosis and treatment methods for pediatric rare disease. Here, we want to combine the basic research and industrial methods. Here, in the Rochberg Institute, in total, we incubated a dozen of companies. They are all the number one company in their own class, including the #1 gene sequencing machine. Besides, I also established some companies. Today, I would like to share several cases. The Rosberger incubator promoted the first gene sequencing machine, the first human genome, the first microbial genome. More than 10,000 firsts were sequenced by this machine. Later, we developed the first chip sequencer, the Biotorum. Nowadays, 60% of the clinical sequencing happen with the ion torrent, but those are all gene sequencing machines. Recently, we developed the protein quantum sign, the quantum sign, the protein sequencing machine. Additionally, another new company from the Rosenberg is Butterfly Network. You saw that you see this name is called Butterfly Network. It's a rare disease. So this is the first one.
声波仪
By using the chips, you generate ultrasound. It's not the previous modality. There were different probes. The machine was huge. Now it's miniaturized as large as your iPhone. Besides, in 2013, I was surprised by AI, though I am a biologic scientist. And there was a Google project for AI, so I started to learn AI. For the butterfly network, we use AI to make a decision about the image, and this is the first approved by FDA in 2017. 30 countries have approved to use this one. Over 200,000 doctors have access to it. And the stethoscopes can be thrown away, and doctors could put this on their necks. Besides, we have hyperfine. This is movable MR. And what is the real signal for MR? What is the noise? By leveraging AI, we can remove the noise, and you don't have to intensify the signal. You can have a simple coil. This was approved by FDA in 2020. Then next, I have several cases that happened in China. Working with peers from Fudan University, we made first mammalian PB transposon technology. For this transposon, it could carry big segments and it could be used for transgenetic studies. In the genome, it can work as a transposon and could lead to mutations. What's its application? We established this company. The CEO is in the audience. And for conditions RDEB, because there is a mutation between skin and body, those people have pemgoid conditions. If there is a small injury, they have inflammation, and there is no treatment. It's a big gene, and it's 9 point something kb in the cDNA, but the transposon can work on it. Our transposon can carry 200 kb DNA. We've got IND for that. Those attempts could help the patients with monogenetic mutations. We'd like to provide new therapies with such platforms. In this Jinshan ACE, we've had CAR-NK. CAR-T is very effective for leukemia. But it works for leukemia very well, but doesn't work for too many other cancers. Why it's not working in some other cancers because you need multiple targets. If you have transgenetic engineering, efficiency and cost are problems. But PB transposon could bring a lot of targets in, and besides, it could bring cytokines into the body, and it could stimulate the survival of NK cells. This can be done in one instance. So we are doing research with CAR-NK. We've got great results in IIT. It's really surprising. The breakthroughs in those methodologies can bring good news for many rare diseases and other diseases. Next, we use PB transposon to have genetic screening to find new drug targets. In the previous genetic studies, you will see which gene is related to a pathogenesis. This is genetic screening. Which mutation leads to a disruption of the process, do a study in that gene. For us, in drug and farm, we go one step further. We look at the new targets through genetic screening. In the animal models, you see a mutated gene, and you find a target. Because a mutated gene avoids the disease, and you can inhibit the derivatives of that gene so that you avoid the disease. You can use antibodies to bind with the receptors. It sounds simple, and why nobody paid attention to that previously? You know, for the previous genetic screening, it was in the low-level animals like yeast, worms, fruit flies, and zebrafish. But for drug targets, you need to leverage mammalian animals. If you have genome mutation for mammalian, it's too expensive. For one or two genes with CRISPR-Cas9, it works very well. But if you induce mutation for all genes in the mice, it's about $1 billion. And it will take 10 years or 20 years and hundreds of people, which is too expensive. By using PB transposon, and we have spent 10 years on it, and in drug farm, we can find new targets. And this is 100,000 USD, and one person, one patient. And you know, for the first time, we can find new targets. We found dozens of novel drug targets. And this was granted to a patent by the US Patent Office for the first time. Then, using AI, we look at those new targets, even they don't have a protein structure, and we design the new therapeutics. It's really surprising. 2 drugs of ours now are in clinical studies. We passed phase 1. The first one, you may guess it. It's for rare diseases. It's for Rosa. It's a really rare disease. In the US, they have less than 1,000 patients. Happily, I tell you this. We passed phase 1. FDA approved this designation as an orphan drug. And this news just was put to us recently. We have high expectations to push this forward. As Dr. Harvey Lodish mentioned, there was the Orphan Drug Act dated 1983, and there is a clause about early license for approval. If you get that coupon, this is worthy about 100 to 350 million USD. We have high expectations for that. Besides the first one I mentioned for Rosa, there is another one for the similar target. If you activate this target, it could reactivate the immune system to inhibit viruses. This is about curing hepatitis B. We've passed phase 1 for safety study, and in HBV patients, we've seen efficacy. It could effectively inhibit HBV DNA, including CGTN. We have 100 million carriers of HBV in China, and we have high hopes for this new possible drug. This is first-in-class drug, and this is now clinical study in the world. We passed phase 1. Dr. Qu Chong, on 25th, we'll talk about this case specifically. I will skip the details. Then, moreover, what are the pending issues to be tackled? A lot has been said about the issues. What is the biggest challenge? Less patients, smaller market. These are big challenges. It's the common challenge for everybody, as Kevin Huang said. You know what? We have a big population. We have an aging society. Expenditure in healthcare will grow. This is a challenge for both China and the US. The expenditure in greater health is 18.2% of US GDP. If this is the trend, they cannot bear the cost in the US. What can we do? We need innovative solutions. We have new ideas. What can we do? Leverage the natural therapies. It's about herbal medicine. They have lower prices, which is a huge advantage. Furthermore, in China especially, there is a huge accumulation of experience. There are over 10,000 monotherapies. This started from the Grandmaster of Zhang Zhongjing. There are 314 formulations, and there are over 1 million formulas. But what about now? There are less which are approved. And this is about 1,607 that have been approved because there are limitations. What are the limitations? Number one, quality control is difficult. You don't know which ingredient is playing its role. How do you control the quality? We don't know the MOA in a concoction. Right. These are challenges. Then what can we do? I believe to turn those treasures into useful therapies so that it is accepted by physicians and patients. We need to leverage modern technology in biotech and biopharma. AI biotechs should be used for TCMs. This is about standardizing the information. Professor Tu Youyou's team have artemisinin to treat malaria, and there is a herb which contains artemisinin. And we have established the systematic nomenclature for NMMs. There are 6 components for any natural herb. It could be accurately named. And we have used AI, and for 14,000 mono herbs, we put them into the database, and it's both Chinese and English. This is accessible to everybody in the world. And there could be chat. Through chat, you can interact. And if you have new information, update that into the database. The TCM is introduced to the world, and the global researchers can leverage herbs for better purposes. Then how to have quality control, how to know the MOA of the herbs. Let me give you one case. I work with Professor Jonathan Rosberg. This is an incubation case. This is AI therapeutics. This is the first using AI to look at new indications. Here's what we do. The genetic sequencing of a patient have changes because they have mutations. Then for different patients have the genetic sequencing, they may have variations among them. This is the profile of the mutations. We make Western medicine. See how these Western medicine change the expression of a gene. If a Western medicine changes the disease in a reverse way as a mutation does, then that could treat the disease because it corrects the disease course. Through the traditional statistical means, it was not working properly. There is around 30,000 genes. AI has its role to play. For AI therapeutics, for the first time, we use AI to have predictions after learning. When AI is trained, this was using the convolution network or recurrent evolution network. We've used multimodality autoencoder, and we used hundreds of thousands of people's gene expression, and we trained that tool in AI therapeutics. Very quickly, we could predict 4 indications. 2 are in phase 3, 2 are in phase 2. In April last year, for ALS, we had 0 drugs, but for ours, we predicted it and we proved to be efficacious in phase 2. Now it's in phase 3. By using AI, by looking at genetic expression, we can predict the indications via AI. And we can do that for TCM as well. For traditional Chinese medicine, we don't know which herb is taking effect. But if we know how it is changing the genetic expressions, we can know how it plays role. Therefore, we've had systemic AI, and we've used big data to train this AI. We built this platform. We could know how the herb is taking its role, how to have quality control, and we predict its possible indications. It's really exciting. It's about combining AI and large data model. You learn about large language models, right? OpenAI and what have you, ChatGPT and ChatGPT-4, etc. Ours is for therapies. Why this is exciting? Because big pharmas have compound libraries. It's about 1 million to 10 million. That's the chemical structures. But for the natural herbs, you know how many chemical structures? It's 10 to the power of 60. There is incredible complexity. Previously, we had no means to look at those complexities. But now, through AI, we can look at the natural herbal medicinals, and for the first time, we put that into a big database and make predictions. The results surprised me, at least. It makes sense to do so. Let me give you one example. For the fatty liver disease, there is no good drug to treat fatty liver disease. By using AI and big model, we predicted one natural herb, and we tried it out in mice. You see those white fat cells after taking TCM by the mice, fatty liver disappeared. And we found a small molecule that is playing a role. How did we find that small molecule? Let me give you one story. Double Rainbow Bioscience is a joint program in Boston I co-built with an MIT professor. This is a company finding effective ingredients in natural herbs. we have extracts, and we run them, and for each black dot is a molecule. Which molecule is taking a role? Previously, you separate them, try them out. It's about tens of thousands. It was too complicated and took longer time. It was costly. How? AI has a role to play. Before AI was adopted, Tuyu's team got to know artemisinin. Within the grass, they tried to find artemisinin molecule. And you need to synthesize that. And that took them 30 years. But with AI, for the quickest case, it took us 3 months. We've got a seed plant and we got that molecule. It's really exciting. After getting that molecule, it was about synthesis in the past. But for natural herbs ingredients, it's hard to synthesize. Maybe 100 will, chemists will take 3 years to have synthesis. We don't have to need to do so. We have herbs. We can get enzymes and get the genes in that plant, put that into the yeast and do it. And it happened. We use AI and genetics to do so. And we made breakthrough in the methods. So this is for the CGTN. This increase the blood perfusion in the brain. And this will increase oxygen supply to the brain at high altitudes.
This molecule is very hard to produce, so we found this whole enzyme and put it in the bacteria for fermentation and production. So in August 2022, this product was approved by FDA and for commercially sale. In the future, regarding the TCM, for the molecules we found in the herbs, in the natural plants, we could use method to develop. So I'm so excited today. When we use AI, use genetics, use the modern biomedicine approaches to combine them with the crystal, the practice crystal from the traditional Chinese medicine for thousands of years, we could help the R&D for rare diseases and other drugs. We have established a lot of platforms. Finally, I would like to talk about our study parameters. There are 3 parameters in the universe: space, time, and mass. Somebody has found the space positioning system. As for how to perceive time, the scientist was awarded for the Nobel Prize. But how to perceive mass? So during the COVID-19, I got no meeting to attend. I reestablished my lab in the Westlake University. I encourage my graduates to do that. So Peng Jikan and Xu Chenshi, we built the model, established the animal models for perceiving the mass. We found the brain area here. We established mathematical model. What were we doing? We were decoding the neural network because the optic neural work was the origin for the deep learning of the AI. So for the mathematical brain area will give us a brand new AI. So the kids in the labs, they've been decoding this one day and night. They want to become the first one in the world for decoding this network. We were looking for the genes. So because we know which area is focusing on the mathematics, we do the single cell gene sequencing. More than 500 mathematicians, they're artists, they donate their genes for us to find genes. We found genes. But what else could we do? What else could we do? We could screen drugs. Because we have animal models, we have dosage models, we have assay. We could do drug screening. We could tell us that we have screened the drugs. All those drugs could help the rare disease to develop, to help the rare disease kids with retarded development. It could help elder in the future. Well, of course, my friends told me, so when you may give me this drug because it would help me for examination. I said maybe we need to have a reconsider for this drug. However, for rare disease studies, what are the challenges? We could use our wisdom. We could do some unique task. This task not just work for rare disease, it works for everything. Thank you. Okay, thank you for your excellent presentations up to now. All the keynote speeches have completed. I believe just now, with all those excellent presentations, we all learned something. Even we learned some ideas and some inspiration. So next, there will be more ideas, which is a great session. It will be a roundtable discussion. So today there will be two roundtable discussions. The topic for the first one is the global challenge, how to increase the rare disease scientific research, clinical translation, and international collaboration.
Inspiring and educative speech. Here comes the first round of our panel discussion, themed Global Challenges, How to Strengthen Research, Clinical Translation, and International Cooperation on Rare Diseases. So now let's give a big round of applause to welcome the moderator, Mr. Gao Guangping, and our guests, Mr. P.J. Brooks, Mr. Chen Wei-yi, Mr. Fan Biao-gao, Mr. Lin Kang-li, David Pierce, and Mr. Daniel Sherman, and Ms. Wang Yi to come onto the stage for the first round of our discussion. 让我们有请本场圆桌的主持人。
Now let's welcome the moderator, Professor Gao Guangping, and panelists for this session. Mr. P.J. Brooks, Mr. Chen Wei-yi, Mr. Lin Kang, Mr. David Pierce, Mr. Daniel Sherman, and Ms. Wang Yi.
Thank you. I think 对不起，我讲中。
Sorry, I speak in Chinese. Sorry. 我想啊。
And also David and Daniel, please excuse me for majority of the audience are non-English speakers, so I think I probably may have to use Chinese. Thank you. OK， 罕见病这个事情啊。
The rare disease, actually, globally speaking, this is a major challenge regarding the diagnosis mechanism or the drug R&D. There are a lot of bottlenecks. This is a global challenge. So human being during the medical development, this is a major bottleneck. So if we want to if we want to make some new breakthrough, the global collaboration is very important. So today we have invited experts from Europe, US, and Hong Kong and mainland China. Some of them, they are experts regarding the basic scientific research. Some of them regarding the regulatory science, regarding the regulation, or regarding the national organization, or regarding the rare disease R&D. They are the leading person, so discussion is quite important. So I hope their views could represent both East and West, China, Europe, and the US and Hong Kong, regarding the institutes and governments and so I hope we could have some discussion. The first panelist is my old friend P.J. Brooks, Dr. P.J. Brooks. So back in 2008, I knew him By then, I just arrived at Massachusetts University. We had a collaboration. It was cocaine syndrome. I don't know if you have this disease in mainland China. So I knew him. So for so many years, we had collaboration regarding the scientific research. But what I admire him is that regarding the rare disease study in the US, for his position in the NIH, actually he has been the promoter for the rare disease study in the US and a key leader. Maybe we know there's a baseball gene therapy from the NIH. Actually, I knew that for precisely. So PJ himself, with his own efforts plus the support from NIH leaders, they created this project. Nowadays, this one created a nation and a private foundation and foundation collaboration successful paradigm. And also, he also lead the somatic cell gene editing. He's a leader. So I myself, with his great support, I got some grants in the last years regarding the somatic gene sequencing. I had some engagement. So first of all, my question goes to PJ.
我应该问中文还是英文 ？Anyway, PJ, I think the first question I will go to you, and as a leader in rare disease research and translational research, and particularly as I just told everyone that in the past decades or so, you spend a lot of energy and efforts in leading rare disease research at NIH, as well as in America, through the collaboration between governments, foundations, and families, patients, as well as pharmaceutical industry. So you have really developed a system. From my point of view, it's really innovative, but also very powerful. We just want you, please, if you could share with our audience, what's your thought and how you think the next step, or how to accomplish what you have accomplished today, such as a bespoke gene therapy and other, I'm sure you still have a lot of thoughts how to further develop rare disease gene therapy to benefit patients and the family. Thank you.
Well, thank you, Guangping, for the kind words, and it's really an honor to be here, and I certainly want to thank Kevin and all the organizers for the opportunity. I've been very fortunate at NIH to have the support of many other NIH leaders to be involved in the Bespoke Gene Therapy Consortium and the Somatic Cell Genome Editing Consortium, and the latter with funding from the Office of the NIH Director. And obviously I work with many other colleagues to make these programs run, so I don't want to give the impression it's just me. I think that as I've been in this field and started with looking at a single rare disease that affects probably less than 100 people, what dawns on me is that the most common form of rare disease that we have is single gene disease or monogenic disease. And the good thing about monogenic disease is we know what the problem is and we know what the solution is. The problem is the one bad gene, and the solution is to get a working copy of that gene into the right cells at the right time in disease development. If you can do that, you can treat, prevent, or even cure the disease. So in a sense, I try to group things together and really focus on that. What are the commonalities across diseases? And all of the programs that I'm involved with, the ones you mentioned, all the ones that we fund, we don't fund anything to do one trial for one disease. Everything we do is about a platform or focusing on commonalities, and I believe that's ultimately the way to get to all of the rare monogenic diseases that we have. And I think in particular genome editing is a great opportunity where you can develop those platforms. And by an interesting coincidence, one of the people we fund to do some of the most interesting research is Professor Yongli Jiang, who's here, and he will tell you about some of the work we're doing to try to develop those. So I think increasingly focusing on therapeutic platforms and commonalities across diseases is the way to impact rare diseases most greatly all across the world. A gene is a gene. The problem's the same, the solution's the same, and I think that's the way we have to think of it.
Dr. Brooke. So, 我的下一个问题是
My next question is for the Professor Chen Wei and President Chen Wei from Hong Kong University. Hong Kong, we know, is an important bridge between Chinese culture and Western culture. Culture, technology, science, in all walks of life, Hong Kong is an important bridge. As one of the bridge, Hong Kong University, I would like to listen to Professor Chen regarding the rare disease development, study, and the drug R&D. What ideas, what concepts do you have, especially nowadays, rare disease calls not just the attention all over the world, but also in China, due to Mr. Kevin for the last 15 years. His insistence on the rare disease and public education and with his efforts. So Hong Kong as a bridge, what kind of roles do you think you should play to promote the development of the rare disease drugs? So thank you. Thank you for having this opportunity to join this meeting. Today I learned a lot from the previous speakers, so I would like to say thank you, Kevin, for organizing this event, and thank you, Talia. So he invited me here, so many thanks. So today, I think your question was not only issue between China and West, or the issue between rare disease or other issues. So regarding the drug R&D, there are several key steps. So the money used for the scientific research, who are willing to give money to do this, because from doing research to having a result, there's a long way to go. So how to focus on this? Secondly, how to invite people with specific skills. I'm from Hong Kong Chinese Hospital, so how to invite scientists with this specific skill to do this study. Thirdly, we all know, so professor is okay for clinical study, but it will be an issue for professors to develop a company. So there's a huge gap from our scientific research to developing into a product. So now we are doing an innovative drug institute. We are planning to have a larger institute. But the issue is, just like I mentioned, first, so how to get that much money, then how to employ that many people, then how could guarantee that we could translate our future results. Nowadays, if you could not translate your results, then you could not afford the sustainability. So finally, I believe that if we could solve this issue, there is an easy way, the so-called corporate lab, just like Kevin just mentioned. So could we attract the pharmaceutical R&D company, attract them into the universities to build their labs in the university? They could bring money, they could bring talents to the university, so we could work together. On the other hand, because they are companies, so they know how to do business, so they could train our professors to do business. And to be frank, in Hong Kong, there are not many large firms. So many companies contact us are from foreign countries, also from mainland China. Because Hong Kong is the bridge between the China and the West. So we could easily attract foreign pharmaceutical companies to do something in Hong Kong, to establish a lab in Hong Kong. Then we could translate it if there is a result. because it's Hong Kong, and then they could go north as well. So by doing this, I think your questions could be solved. So the company money are to develop drugs, right? So this is currently, this is my, so corporate lab I think is quite important. I think large companies you may consider, like for example, they could have like a title for the rare disease. It definitely works. Because for rare disease, it's really difficult. There are not many investors, but if you want to do that, you could make use of our staff. It's easier. So I don't know if I answered your question or not. So excellent. So thank you, Professor Chen, for your insights, for your sharing on the ideas on developing the R&D of the rare disease drug. Thank you. Next, I would like to introduce my colleague in the Massachusetts University, Professor Gao Fenbiao.
先后1年。
Professor Gao and I studied MIT research 1 year and but another, and but there were missing emails because our colleagues could not distinguish me and him. But we do different business. For genetic therapy, our treating of rare diseases, there are 4 components. Number 1, get to know the genetic mutation and also pathogenesis. Secondly, have a good delivery system so that genetic therapies can be injected. Number 3, there should be a good animal model to validate the genetic therapy for rare disease or otherwise. And number 4, how to deliver the medicine. For different conditions, there are different deliveries.
呢是
Professor Feng Biao does research into MOA or pathogenesis. So for the platinum studies and translational studies, they provide adequate, safe, and right targets. In the meantime, Professor Feng Biao established many animal models, and these are instrumental for genetic therapy. So, would you like to share something about your experience and your ideas? Thank you. Okay. First of all, it's great to be here to see old friends and new friends. Mr. Huang has given us a very wonderful keynote presentation. In one of his slides, he put scientists and research as the center. Fundamental study scientists are important in the first steps. I have Peng, Xiaojiang, who are friends of mine. For our approach, it's quite similar. We have different cellular models, animal models to do studies. This covers the worms, fruit flies, mice, pigs, and the rhesus monkeys. With those animal models, we look at the basic mechanisms of mutations. Such studies are very important. First of all, we need to know after mutation, is it a loss of function for that gene, or is that a gain of function, which is a toxic function? If it's a loss of function, we can use AAV to increase the expression of the gene to restore the function. If it's generating toxic function, we can use RNA technique to knock down their gene. Those approaches can possibly be a treatment approach. The other thing is to look at the lower stream mechanisms. You may not be able to crack that mutated gene, but if you know the lower stream mechanisms, you may change a lower level gene with small molecules and other approach. so that you could treat the symptoms. Fundamental studies are very important because such studies can provide new targets. Thank you very much, Professor Feng Biao, for sharing. You've got great results and experience in this regard. Next. So, the chairperson for China Rare Disease Alliance, Professor Li Linkang, you know, this is one of the co-hosts, and China Rare Disease Alliance have been promoting the industry like diagnosis, treatment, R&D of drugs. And Zhang Shuyang was here, and PUMC Hospital and PUMC worked very hard on this as well. For rare diseases in China versus rare diseases in the world, of course, we have big patient populations. Professor Li, what are your considerations? Or from a national policy, or from the China Alliance for Rare Disease, how do you support the research in rare disease, the research, the diagnosis, and treatment? Thank you. I thank Kevin for invitation. After hearing the keynote presentations, I've learned a lot. For rare diseases in China, with the support of the government agencies and stakeholders' efforts, diagnosis and reimbursement is growing, especially R&D of drugs. Our alliance have been working for 5 years, either for lobbying in policies or improving the capacity of doctors' diagnosis and treatment, or working with advocacy groups to promote the awareness. A lot of work has been done. However, Professor Harvey mentioned, if you want to specialize in rare disease, have the passion, have the companies that make money. The policies are being pushed forward in China. Diagnosis and treatment is being advanced, but there is a big gap between what we have and what we hope to have. R&D by biotechs are not high because the policies is not mature. There may not be reimbursement if a therapy is launched. So biotechs are not willing to invest too much. For the alliance, we plan next step for this. We train doctors to improve their capabilities. 5 years ago, I was starting this alliance. You see, we had less physicians, rare physicians who are rarer than the diseases. Now we have more doctors who can treat rare diseases. Now it's about more investment. For the alliance, we step up our efforts for training physicians for rare diseases. And in lobbying reimbursement, we are doing our part. Looking forward, with the Chinese economy growing, with the efforts of doctors, we'll have better coverage and we'll do a better job. Thank you. Thank you very much. Professor Li, what you mentioned is very important. It's about the Chinese policy framework, the Chinese society, biotech, biopharma, hospitals are doing their respective parts, either making policy frameworks, improving diagnosis and treatment. Yes, this started from zero, and this is growing very quickly. We're happy to see such progress. In the presentation by Kevin, Mr. Huang asked what we can do in China for rare disease. That's well said. Very good points. One of the points he mentioned is that collaboration. When you come to collaboration, we have international organizations like ERDIC. We have Professor David Pierce with ERDIC. From your perspective,
the bottom line of the organization you are leading or you direct is collaborations. And also, very importantly, international collaborations. That's why you are here. That's why we have this meeting in Shanghai. And so I think this is not only international, but also in each country, different scientific expertise and also different entity, government, foundation, family, as well as the pharmaceutical industry, all need to be collaborative to develop drugs for rare diseases. So if you could share what's your thoughts and particularly the mission of your organization and how approaches and accomplishments you have in the past decades since establishment of this organization. Thank you very much.
Okay, so once again, thank you for the opportunity to speak to you here. So collaboration, there's a shorter word that really describes collaboration. The word is yield. And what do I mean by that? I mean the fact that right now on this planet, there's maybe 300 individuals that we speculate have a rare disease. The human genome, we estimate, is about 3 billion letters. Any change, whether it's monogenetic or potentially haploinsufficiency, in that 3 billion letters could cause any cell type, and we have many cell types, or any organ to not function correctly. So when I say yield, no individual or set of individuals can understand all of the consequences that I just explained. So the International Rare Disease Research Consortium is a conglomerate of individuals from around the globe that have expertise in their very specific areas. So if you listen to others, you will learn all the time. So how did I get to be the chair of ERDIC? So I'll give you a couple of examples of how yielding and collaboration and then moving forwards with respect to that. So I'm a basic scientist and I was working on a lysosomal storage disorder called Batten disease. You create a mouse model. Then you listen to someone who tells you, mice really are not very good for studying vision loss because they don't have a macula. Their eye is not the same as a human eye. Let's build a pig model because the pig model is going to be much better to study the eye. Batten disease is a very slow neurodegenerative disease. Let me listen to some of the physicians and patients, or the families of the patients. What are the observations that they see? Because they live it every day. So the first clinical trial that was ever developed for Batten disease was for the fact that there was a novel symptomatic autoimmune component that was contributing to the disease. That was listening to others. So the International Rare Disease Research Consortium is this, as I just said. So we have, I don't know how many countries are in the world, but I do know there's a lot of different countries. And when we get together, we talk about, well, what does it happen in this country? How do you do it in that country? What do you do with this disease? How do you do it with that disease? So an example is we have two special issues in the Rare Disease and Orphan Drug Journal right now on newborn screening. Two special issues. One which is really just talking about some of the technological developments, because guess what? In different countries, different platforms for diagnosis are being adopted for newborn screening. The second issue is about policy and ethics. When we would discuss this within the room where we had people who are neighboring countries in Europe, the ethics and the rollout of newborn screening is different within Europe. So how do you listen and learn on how you do that? And obviously, culturally, that's very different across the globe. So the point being here again is that I'm a basic scientist and now I'm involved in economic analyses of the impacts of the treatments that could be developed for rare diseases. So it's a wonderful thing. So I would encourage you, every time you're at a rare disease meeting, and if you ever engage with ERDIC, we have people that study every aspect, right from that single cellular defect, that single base pair change in a genome, all the way up to Why does it cost $2.1 million to treat a child with spinal muscular atrophy with the cutting-edge gene therapy that came there? To then also going to the patient advocacy groups and saying, you know what, you've obviously just saved that child's life. They're going to have a meaningful existence. And also, guess what? One of the parents now can go back to work. Both of the parents aren't actually taking care of that child constantly. The global impact of rare diseases, I think that no individuals can really understand all of it. So that's what collaboration is about. It's about yielding and listening to the expertise of those specific areas.
Thank you. That is perfect. That's what we're looking for, and that's the whole mission and objective of this conference. So my next question will go to Professor Daniel Sherman. So I actually have two layers of the question. First, as a gene therapy researcher, a well-established, a well-accomplished gene therapy researcher, and we would like to know if you could share some experience in terms of gene therapy research community in France. What's your thoughts? How do you develop gene therapy for rare diseases? And what you have done, what you accomplished, and what are in the pipeline? Second is, as the Rare Disease Foundation chairman, and how disease foundation families could play a role in rare disease gene therapy or diagnosis and basic research. Actually, myself, this question is highly relevant to myself as well. I went to America back in 1988, trying to learn next generation of the medicine. I started rare disease research in 1989. So what I really feel, my professional growth, my scientific growth, is really highly associated with patient foundations, organizations with patients. So I truly feel like during my years of rare disease research, I met many families and visit their home and interact with many patients and parents. I really feel like they are like our motivation, our engine. So at this point, I really want to hear from you what's your thoughts and in those two areas, scientifically as well as foundation-wise. Thank you.
Well, thank you very much, and I would like to thank Kevin for this extraordinary meeting, and thank you for your question. So the first part is about my scientific opinion and view. Mainly, I've been working since the last 30 years in the field of non-viral gene therapy. And this is a very interesting story because at that time we could not handle mRNA. So we were using DNA, and we developed lipid nanoparticles in order to deliver this DNA. It happens that lipid nanoparticles is very efficient to deliver DNA in solution, in vitro, because you have cell division and the plasmid can travel to the nucleus. But on post-mitotic resting cells, the plasmid cannot migrate to the nucleus. So it was not so efficient for in vivo gene therapy. So we shifted to a physical technique, and I invented a technique whose name is electroporation, which is able to deliver a plasmid into 100% of muscle cells. And electroporation now is used in vitro for transfection of, for instance, T cells for making CAR T cells. Electroporation can be used to deliver plasmid and to incorporate plasmid into the genome the same way as lentivirus do by using a transposon. And we had a presentation of transposon, and I am going to present more in my talk Saturday, where virtually what a lentivirus can do, you can do exactly the same for a much lower cost by using simple plasmid and electroporation. But now we can handle mRNA thanks to the recent discovery. And these lipid nanoparticles became an incredible tool because with mRNA and with CRISPR-Cas9, we can use a single shot, short half-life gene tool. So now we can deliver with a lipid nanoparticle an mRNA to the liver, for instance, and this makes gene therapy, for instance, for transthyretin amyloidosis, with a single shot of a lipid nanoparticle containing an mRNA that will do the genome engineering and that will destroy the mutated gene because it's a gain of function mutation, you can have a treatment for life and for very, very, very cheap. So, I mean, there are room for viruses, AAV, lenti, there is room for non-viral. Another aspect of non-viral is siRNA, small interfering RNA. And I've been also involved in the delivery of siRNA, several patents. And what we found that siRNA were short-lived. We were looking to siRNA delivery to the liver against hepatitis B. We knew there was a problem for chronic infection with hepatitis B. And when we deliver the siRNA to the liver, we have 2 to 3 weeks silencing of the hepatitis B gene. But now with the new discovery of chemistry, there has been so much, I would say, improvement and progress in the chemistry of siRNA that now with a single shot you can achieve from 6 to 9 months of duration. So it's once a year or twice a year treatment. So this is what they are doing now for transthyretin amyloidosis. For hepatitis B, that could be an interesting opportunity. So what I wanted to from the, let us say, scientific point of view, think viral, think non-viral, be open to any technology. For instance, we are now developing plasmid gene therapy with an Australian team in Sydney, where they deliver a plasmid for a neurotrophic factor in the cochlea, and with our plasmid, they induce a regrowth of the nerve, of the cochlear nerve, acoustic nerve, and they reintroduce hearing into people with complete loss of hearing, and this now will start to phase 2. So we have some quite interesting progress with this kind of new technology. It's electroporation and a mini plasmid engineer in our lab. Now, in my second life, as a head of the Foundation for Rare Disease, what I would say is that, first of all, about collaboration. China is huge as Europe, I say. What we have at the European level is what we call European Reference Network. It's a network of reference where specialists in a specific type of family of rare disease, we have 23 different families of rare disease, they coordinate themselves and they meet on the teams or every twice a month, and they discuss cases. So they, it's international, and it could go with China as well. I mean, specialists from all over the world have a monthly meeting with the present case, and they say, what do you think? And it works very well. Because for some disease, maybe you have 1 or 2 specialists in France, 1 in Germany, 1 in Italy, and this has been going on very well. You need some incentive, money incentive for make it work. About diagnostic, I would say that we have supported a system, artificial intelligence system, to help general practitioner to identify a disease. But not to identify a disease, to help him to identify that this might be a rare disease and to direct it to a reference network. So there is a complete, channel of help for the general practitioner to identify that this is a rare disease, to classify which kind of rare disease it is, and then to guide him to send the patient to the specific reference center. And then towards this European reference network, they coordinate together for the best diagnostic. So you have a lot of progress. What I can say, our foundation is not a patient advocacy foundation. We are a foundation to help science, to support science. We deliver money to laboratory. but we also to help patient advocacy organizations to do their science. We advise them, we help them to put on a scientific committee, we organize calls for projects on their disease, they bring us the money, and we treat with the university, and we follow and we monitor the project. So we are some kind of scientific coach for about 170 patient advocacy organizations. I think this kind of organization can be very helpful to promote and to foster research because, as it was said, patients are very important for the starting of research, but patient advocacy organizations, sometimes they need help from professional scientists, and this is what we are in the foundation, to give them on a free base, it's a service for them, to help them to do their science and to promote their science. And finally, what I would like to say is that, because it was mentioned, you should think about repositioning. Repositioning is quite an important, and we had a presentation of the TLC signal transduction with the TOR and PCSK and PIK3CA. And you know, this has been used, some drugs have been developed to treat cancer. It was mentioned that 70% of cancer go through there is mutation in this transduction signal. Now, this drug have been repositioned to treat overgrowth syndrome, which is a rare disease, because it is initiated, it is caused by the same PIK3CA mutation than in cancer. So you can use drugs that have been developed for large disease like cancer, and you can try to reposition them to rare disease, and this can be very helpful and I would say a short way to go for treatment for rare disease.
Thank you very much. That's fantastic. My final question. 对不起，我最后一个问题。
Sorry, my last question is for President or Professor Wang to share with us the Fudan University Children's Hospital regarding the rare disease study, regarding the study type physician team building and as a research type hospital building. We know Fudan and Xiehe, they are the leaders in the rare disease in China. So your experience, I think, is very important for all rare disease colleagues. So please, okay, thank you. Many thanks to experts for coming all the way to Shanghai. I think just now I learned a lot from your speech. So our children's hospital is the National Children's Pediatric Center. We could use 8 Chinese characters to show our work or course regarding the process learning. Our center regarding the diagnosis and scientific research. Actually, we need to learn from all over the world. We started late. However, we had good clinical rare disease resources. So at the very beginning, regarding the pediatric rare disease, we have been following, we have been learning from others, especially for some pathways at the very beginning, back in 2016, in NIH, guided by Gold, we established the first UDP center in China. So actually, regarding some specific projects like the EPI-25, like the epilepsy international, like the Angelman, every year we have Angelman conference, like for the MDA conference. So to gather the information in those fields. Actually, we have been participating, for example, the American UDM meeting and the ERDIC meeting. So during those meetings, we may know more experts. We would know their key focus. This is my first keyword. My second keyword is participation. We, Fudan University affiliated Children's Hospital, we've been participating multi-center rare disease clinical trials for many of them. For example, I joined the recipe plan from the Roche focusing on SMA for the fabric disease and other disease, and some allergy or the genetic liver disease to join the international study. So our team was trained, the base was built. Meanwhile, we could fully participate the global new drug R&D, the gene therapy. So I think this is really a good training and learning and the participation and following. Third keyword, I think it would be sharing. Actually, I think we have a lot of rare disease organizations like the Kevin together for many children or pediatric rare diseases. We have close correlation, close cooperation with international organizations. We have relevant teams to develop some cutting-edge studies. So this is really a good sharing. And also, next, regarding the single disease cohort. For example, we are building some RBSC and organoid bank, which are we have abundant genetic mutation cell models and like some monkey models. Those are all good resources which should be shared. We should share with them with other experts. The fourth keyword would be the collaboration. Regarding the collaboration, established you just mentioned, we need to internationalize our resources, our scientists, including the talent cultivation and scientific research collaboration. I think with our continuous promotion, definitely in the field of the rare disease, together with our colleagues, we could learn, we could share, we could collaborate with everyone. So thank you. Thank you, Professor Wang and President Wang. Yes, your four key words is quite meaningful for the rare disease community, and I would like to say thank you to all experts who are present here. So actually, we effectively discuss regarding the rare disease, R&D, and the study, the key pain points, which were mentioned in Kevin's presentation. And from a scientific perspective, we had in-depth discussion. I hope this roundtable discussion will be helpful for all of you, will be helpful for the career of the rare disease career in China. Okay, thank you.
And the insights, please go back to your seats. We really appreciate it. Yeah. This way, please. Thanks for your time. 感谢各位嘉宾的
Thank you, experts, for your excellent discussion. I believe that today, with the discussion just now, we all learned a lot. Next, let's move to the second roundtable discussion. The second roundtable discussion is the topic is that how China will be integrated in the global rare disease drug R&D.
distinguished guests. They are Mr. Kevin Huang, Mr. Yonghui Zhang, Mr. Li Xiaojiang, Mr. Harvey F. Lodish, Mr. Adam Risnick, and Mr. Xu Tian, Mr. Zhang Xue to come onto the stage for the second round of our panel discussion. So previously, we've talked about the significance when it comes to the global cooperation in promoting the research and innovation in the treatment of global rare diseases. And now in this round, we're going to focus more on China's role, how China can integrate into the global rare diseases research and orphan drug development. And we really look forward to the second round of discussion. All right? So now the floor is yours. Thank you.
Thank you, moderator. Let's make sure that our foreign guests could have simultaneous interpretation service. Okay, for this session, I will serve as the moderator. I will be serving as the moderator for this session. I'm quite interested in this topic. And this is also I really want to talk about regarding the global research, regarding the R&D of the rare disease drug. What role could China play? During the last panel, we discussed the global collaboration. For this session, let's focus on China's role. Well, 6 panelists for this round. So, besides Professor Zhang Xue, he lives in China. Professor Xu Tian came back as well. So, all experts, actually, you all had a lot of collaborations, including Mr. Xiaojiang now is back to the Jinan University. So today, I would like to invite you to have a discussion on this topic. I think each and every one of you, you have all the rights to say something. So first, I would like to introduce our panelists. First, Professor Xu Tian, President Xu Tian, and Professor Harvey Lodich. Thank you for your excellent keynote speech. Welcome back. Also, I would like to say welcome my good friend Adam. Adam, due to flight delay, he just came to the scene. He was supposed to be here yesterday, so a long run. Maybe the jet lag is killing him right now, so he could catching us. We still have 2 professors, Professor Jiang Yuhui. I knew Professor Jiang Yuhui a long time ago. Today, we are so happy that we could invite you to our symposium again. Professor Xiaojiang, we know him very well because we have some collaboration projects. We've been discussing, we've been promoting. So finally, I would like to introduce academician Zhang Xue. Academician Zhang, actually, I've been calling him Professor Zhang for a while because 10-some years ago, I knew Professor Zhang. When I started my rare disease career, academician Zhang Xue came all the way to join patients' activities. So today, I'm so happy that I could, as a non-scientific professionals, to be the moderator for this session. So please forgive me for my wrongdoing. So I have several questions. Because Adam was late, so you are privileged. So it's a long time for you. So I know, Adam, in the Philadelphia Children's Hospital, you've been leading a large team. I know. So you have more than 100 PI teams. There are so many study funds in the US, and I know you love China. You've paid many visits to China as well. Except for COVID-19, actually you will come to China every 1 year, every 2 years. So my question to you, you have so many scientific research programs in US, especially for the brain tumor for children. In your opinion, how patients' resources in China, how the clinical resources in China, how the scientific resources in China could join your program? Thank you.
So first, it's great to be here. Like you said, we haven't been here since COVID essentially came. And for me, every time I come to China, a lot of the same lessons that were just highlighted come to mind, which is that China as a country, one itself is very large. In fact, I was telling you, sharing with you that as I was landing, because I was late, I was watching your speech on WeChat. And you had more than 10,000 people watching, right, your speech. That's 2 orders of magnitude larger than any presentation I ever gave in the United States online. And that scale, essentially, is one opportunity that China brings to bear. But the other part of that, which I think is tremendous, is, for me, what's been really exciting to see is the level of connectivity, right? So the fact that you have 10,000 people watching means you're connected to 10,000 people, right, watching. So the other part that I think in China is really exciting to see is the level of intersection between technology and its application. The unification of technology across both mobile phones and WeChat and healthcare. And it's really that connection, right, that provides the opportunity to transformation. And we see that's true in the United States. So across the many programs we run in the United States, it's really connection between information and people that has a chance to transform. We heard a little bit about the Undiagnosed Disease Network a second ago, the UDN. In reality, sometimes in a rare disease context, all it takes is one other patient with a mutation in the same gene and the same phenotype to diagnose a disease. For that connection to be made, technology now can do that faster and better than ever. And so for me, this combination of scaled technology, large-scale data sets, and real-time connectivity sets the stage for transformative impact for the rare disease context. In reality, if you can connect, you're no longer rare. And for me, that's part of the long-term mission of coming back to China, is to really figure out ways to connect across the data, diagnostic, and information landscape as these new technologies emerge, because it's through that connection that discovery will be enhanced. And that connectivity is no longer separated by distance, right? It took me 14 hours to fly here, but I could watch your WeChat instantaneously no matter where I am, right? That real-time connectivity of information on behalf of translation and impact for patients is the global opportunity that we at least embrace.
好的，谢谢 Adam。
Thank you, Adam, for your sharing. I have a follow-up question. You've been building many databases for rare diseases in the US or in the world. How does China fit into your database, for example? And what are the challenges if we do so?
You're absolutely right. I think this is one of the areas of opportunity that I think we need as a rare disease community to continue to explore. And that's because, it turns out that if I see a mother with her son or daughter who's affected by a rare disease in China or in Europe or in the United States, they look the same. But there's still challenges around the flow and intersection of data. And for legitimate reasons, right? Even in the United States, we have to protect privacy for patients. We have to ensure that patients consent to drive their data sharing. And so it's really the challenge that we see, whether it's in the United States or across China and the United States. It's really the combination of policy and frameworks that can harness that technology for impact. So today, there isn't full-fledged communication between rare disease data sets, except when you have direct partnerships like we heard earlier today, right? When you have key stakeholders who lead interactions, right, between communities. But I think there's an opportunity to scale that in a governed and structured way. And I think that's the other layer of technology that people forget, that at the same time that we have increased capacity to connect information, we actually have increased transparency about that process, increased tools to govern, monitor, and assess appropriate connectivity of information. And so it's really trying to bring those two opportunities together that sets the stage for what I think you're asking me, right? Is the opportunity landscape now is larger than ever, right? The amount of data that is generated in China, right? Just like the amount of people watching WeChat is orders of magnitude larger than many other contexts. That's a very powerful framework, right, for discovery if you can establish connectivity and harness that human architecture. And as you heard earlier, and as you highlighted, it's very challenging to commoditize rare disease research, right? So In many ways, it's the rare disease context that's most prone for connectivity, meaning it's the lowest risk to connect across rare disease data. The patients and their families are often very motivated to try and find a cure, so they are consenting participants. The clinicians and doctors who are taking care of them are already part of networks that often communicate, publish, coordinate. If we can match those established frameworks of human participation with technological participation, I think it could be transformative.
好，谢谢 Adam 的这个分享。
Thank you for your sharing. I agree. For rare disease, I agree. You see, there are less patients. They're in different countries and regions. For the data of rare disease research, they could be connected and shared for common purposes. Now, I have a question for Professor Jiang Yonghui. Professor Jiang is a professor in Yale University, and you lead 2 NIH programs now, and these are grants worth of tens of millions USD. From your perspective, I know you travel back to China frequently, and you have lectures in Fudan University or joint programs. So from your perspective, for the US and China, what are the differences to apply for grants? And what are your recommendations for the researchers in China? This is a big question. For example, for the National Natural Science Foundation, they have funding for programs for Chinese scientists. What are your recommendations so that we can have more studies for rare disease? Thank you, Kevin, for your question. Yes, indeed. For my research, I get benefit from foundation or NIH government-sponsored programs. I'm a reviewer for many grants as well. If there are researchers for rare disease, you can say I am an expert in a certain rare disease. If there are only 20 cases, I've got 4 cases already. Or you could say there is no expert for rare disease. You've seen 20 cases, you don't know the disease thoroughly. Then there is a difficulty. In the review, possibly there will be a bias, because this is purely based on your experience. I always tell the junior faculty, be humble in rare disease. When I see a patient clinically, patients always give you surprises whenever you see them. 3 years ago, you saw that patient, and 3 days later, you continually learn every day. They are rare diseases. You've seen 10 cases, and the number 11 could be a surprise. Even you have been learning in the last decade. For China, in the last 10 or 15 years, rare disease research is picking up, and the speed is very fast. You know, for China, we got to know rare disease in a reversed way. When there is a genetic sequencing modality, a doctor finds, oh, this is the condition, and this is the mutation. Compared with my training in the US, I got the forward way in my learning. You treat the patient, you make a diagnosis. But now it's genetic sequencing, which comes up with the mutation. This kid has this condition. Those conditions, most of the time, are not well treated because many specialties are not mature. Physician sees a surprise after seeing a disease because this physician was ignorant before that. It's about naming that condition, seeing the condition together with the family members. Even if I practice clinical genetics for 20 years,
it's still a lot of challenge. Just be humble for the 很多年轻的这个做罕见。
So for many of you who are scientists or clinicians, be humble.
Objective and for you review the grant when you.
好，谢谢江教授。
Thank you for your sharing. A question to Professor Li Xiaojiang. He's taught in Emory University in the US for years. When he returned, he started working in Jinan University. He leads a very important research for Huntington disease. A lot of efforts have been put into Huntington disease. This is a central nervous condition. So, Xiaojiang, could you share some differences about research in the US, in China? What are the different support? I'm curious. I have similar questions to you all. Number one. Number two, Xiaojiang, when you are back to China and doing research, will your translation be more convenient so that you can put that into market? Thank you, Kevin, for inviting me to be here. Great to be a panelist. Your question is really good. Number one, what I feel about China. I studied Huntington disease for 23 years in Emory University. In the studies in the US, I've used mice to study Huntington disease. It's monogenetic mutation. The phenotype is quite clear, and there are many similar studies. The studies could shed light for some other neurodegenerative diseases. I've been doing research for over 20 years, but there is a drawback. You cannot emulate the death of the neural cells. This is not limited to Huntington disease. For Parkinson's disease, Alzheimer's disease, all mice models could not simulate that. So coming back to China, I'd like to use big animal models to explore the possibilities. Recently, we found with big animal models, you can simulate the pathological changes. It's informative for rare disease, but also other diseases. With good animal models, if you have good phenotypes in the pathology, you can screen the right drugs. For dementia, global investment is around 600 billion USD, but there are no effective therapies, and MABs are used. The bottlenecks were small animal models, and they may not have clear depth of neurons. There's a gold standard to assess a therapy for dementia. And then, when I came back to China, I see the Chinese government attach importance to science and technology. And the Chinese government is recruiting young scientists. That's why there is high speed in such development. As you can see, the quantity of young scientists, the papers published, are both increasing rapidly. China is incorporating itself into the studies of rare disease and otherwise. China has its advantages. If you look at rare disease specifically, it's called rare PERSA, but we have a huge population. So for rare disease, may not be so rare in China versus the American population. Our patient population is huge. The second affiliate hospital to Zhejiang University, they have 500 patients with Huntington disease. You don't have so many patients in the US or Europe. So for the population of a certain single disease, you have a huge population of patients. These are good resources to screen therapies, to assess therapies. Another thing, a monogenic mutation could have different levels of incidence in different countries. The genetic background are different. There are different environmental factors in different countries. For the resources in China versus resources in the world, we need to pull them together. so we can find genetic modifiers so that we delay the onset of the disease. And that's important as well for the treatment. So this is first feeling to share with you. Number two, to have fundamental studies in China, there are advantages now, especially for big animal models. China is yielding a lot of good results. In my lab, a lot of the discoveries show that in a mice model, you don't see a phenotype. In a big model like swine or non-human primates, you see such a phenotype. And China needs to share the data with the global counterparts so scientists could have access to these results. We have big model resources. For the city I work in, Guangzhou, have a lot of resources. There are 2 animal farms, and each animal farm have over 30,000 rhesus monkeys. You don't see it everywhere. In terms of animal resources, China is rich. There are advantages. So this is a very important symposium today because we can divert our attention to the studies in China. I thank Kevin for his great efforts. I hope we have more opportunities for exchange. So we step up our exchange with counterparts from the world, and so we can push this industry forward. Thank you, Xiaojiang. You mentioned that China has advantages. I'm happy to see that. We have a huge population group, clinical resources, etc. There's another advantage. We have multiple ethnicities, and they could be good resource for studies, and we need to contribute to the global cause of rare disease. Now, this question goes to academician Professor Zhang Xue. You're a master for genetics in China. In 2016, you got a grant from the central government for cohort studies of rare disease. Now you lead many studies for fundamental science of rare disease. Here's my question. Since we come to this topic about how China integrates into global rare disease research and orphan drug development, so from your fundamental study perspective, could you share something about your progress in fundamental studies? Then, in the future, will you have any candidates for translation studies or for this international symposium as a scientist who do fundamental science? For overseas experts, what have they brought to you, especially opportunities for cooperation? Thank you. Great. It's a big question. Everything is possible for cooperation and communication. I have Professor Tian Xu here, who I admire very much. And, you know, his research field is huge. He has done fundamental studies, drug discovery, and more importantly, he now specializes in study of TCM. I salute you. Great to see so many old friends like Jin Peng, Jiang Weiyi, and Xiaojiang. We call ourselves medical geneticists. If I do not get it wrong, Jin Peng is a PhD, and Dr. Tao is MD and PhD. So we have similar ideas. In the rare disease field, if you look at China, we should actively participate and make our contributions, number one. Number two, and we need to share the results and cooperate bravely. And this is the right direction. For some policies, we may not talk about them, though there are limitations, but we need to take this proactive view. As I mentioned, it's monogenic or single gene disorder. They can be different names, but it could be a single gene. When you look at mutation, phenotypes could be different or the same, and in Caucasians in the Chinese population. In my previous studies, I find one key thing is for alopecia, and we work with scientists from 10 countries to study alopecia. There is Mariana hypotrichosis, and there is genetic hair loss. Scientists from 10 countries have this study together. We pull the efforts together, and cooperation is utmostly important. Secondly, through such symposiums like these, we can learn from each other. We've heard the professor's presentation. We know Professor Harvey Ladish. I know Professor Ladish because I read Nature and Science. I studied in Boston for some years. I had a lab, and it was very near to Boston Children's Hospital. Yes, they do great work there. I could feel the academic atmosphere. They have the mechanisms for translation. In Boston, if you start up a biotech, there is a great atmosphere, great infrastructure. Fundamental studies are combined with clinical studies. There's an ecosystem. We encourage such innovation. Now China is different. We have Tianxu and Jinpeng. You could feel that China is encouraging innovation as well as translation studies. There is a huge change. If you look at investment in programs, as Kevin asked about grants, in 2016, actually, I started as my role in the Dean of POMC in June of 2010. I was studying rare disease. I got grants. And it's from Natural Science Foundation of China.
So back then, during the 863-973, we had such projects. As for Natural Foundation funds, I used to have two key funds for all for monogenetic disease. Now together with Xiaojiang, we are leading the national major project, not that major. It's about 10 million. Then the national key development and the national precision project, and the President Su Ran as the PI. We've been doing this for a long time. However, all those projects could not meet the urgently needed needs regarding the rare disease study. There are so many needs from the patients and the requirement from the government. Nowadays, our team regarding the basic research or the clinical diagnosis with our team has been developing. But the research could not meet all the needs. One of the needs is that we need to really catch up the study work in the developed countries rapidly. Just like Director Jiang just said, it's not like what you said. When the gene sequencing was not available, like clinicians like Wang Yi, they were treating the rare diseases, but they were only relying on the lab tests, or they were also treating the patient, but they were not as precise as a certain gene, like for the Huntington disease. Without gene sequencing, but the clinical diagnosis could be made, like the ALS, the clinical diagnosis could be made. But the issue is that nowadays, with the high throughput sequencing technique, now it's more precise. In the past, diagnosis in the grassroots level hospital, they could not make a diagnosis, but now they could. So now we had enormous progress. That the performance of clinicians are different. If everyone could work like the Xiehe Hospital or like the President Wang Yi's hospital, I stayed in the Stan's Chance building. I know the neuroblastoma or other conditions, they were quite good. Patients all go there. So in China, this kind of referral consultation mechanism is not as good as theirs. So regarding the rare disease, we not only focus on the study, not only focus on the drug R&D. Regarding the policy, regarding the diagnosis and treatment, regarding the diagnosis and treatment, and hierarchical treatment, how to share, how to coordinate. There are so many things to be done. So this platform is a good one. So let's speak up together. Let's work together. Okay, thank you. Thank you, academician Zhang Xue. Thank you for doing a lot for promoting the rare disease. Just now, you especially mentioned President Xu Tian. This question goes to President Xu. Thank you for, since you came back China, you started my work regarding the rare disease promotion. So President Xu is different from others. After returning from Yale, President Xu joined the Westlake University in China. We know Westlake University is the top-level research type private hospital. This is a new model in China. I know that the Westlake University has been continuously introducing the top-level scientists into the Westlake. So my question to President Xu would be, what made you go back China and join the Westlake Hospital, the university? And what is the call that the Westlake University could attract so many great scientists? Good question. So Westlake University, just in a few years, yes, we recruited 35 lecture professors and a large number of young but excellent professors. At the very beginning, it was difficult. For example, back in 2019, we gave 57 associate professors. But many of our candidates, they went to the Europe or the US. But in recent 3 years, yes, among our 100 associate professors, this is a global competition. Then they come to the Westlake University. They didn't go to MIT, Duke, or Cornell. Dr. Harvey Lodish
give up some professor offer from MIT to join us. So we very proud and 所以然后我们
Then those 35 lecture professors are really difficult because from the single digit, yes, we have used to have single digit lecture professors came to Asia, but it's really rare for us to gather 35 lecture professors to Asia, to China, UT Southwest, or UC, or San Diego. At very early, they didn't have that many lecture professors. So what made it? I think there are several key factors. Say, we encourage crossover. Then it's really hard to do crossover in old hospitals. If you want to donate a building, like AI is a building and the neurology is a building, they don't talk with each other. But it happens that neurobiology and AI guys, they have to communicate. Then they could come up with new ideas. So it won't happen in the old university, but it's relatively easier in the new hospital, a new university. So we use an academic ring to connect everyone together. So when you get up in the morning, you could see that in seminars in different departments. So when I back to in Yale, I talked with Peter. I told him new universities is is quite attractive. Well, of course, it has a lot to do with the economy development in China. If the China economy is not as good as today, it's impossible for us to attract so many talents. So we are still very careful, trying our best to attract the best elites globally. Here, I would like to please allow me to make an advertisement. So not just the Chinese, we want to join all the scholars.
is essential for 这个整个这个环境。
For the whole environment improve is very important. But back to the rare disease. Since today is a rare disease symposium, I would like to say a few words. I would like to speak in English.
Absolutely amazed to see so many people from different parts of the world come here to China for this rare disease event. It is absolutely fantastic. I feel so good. At a time when we have wars, we have conflicts, we have politicians try to pull, countries, societies apart. And here, people from different world come here for the common good of humanity, for understand and treating rare diseases come together. So I'm absolutely amazed, and I feel so much positive. So I think this is a really great opportunity. And I will call for, say, hey, in this area, I think actually different countries will all agree that we could actually pool resource, pool effort together to do something. This is probably one of the few areas that actually even politicians cannot reject. So I think it would be fantastic. So for we actually, for what China can do, I think actually a group of us should go to Chinese government, say, hey, give us a major grant for this area to support international collaboration. And I also actually called for people from different countries, from United States, from NIH, from different US institutions, to also say, hey, can we actually form some form of collaborations and to really work on rare diseases together? Because obvious reasons we need to work together. Right, and also to include everybody together, the number of patients increase, and also the efforts and resource increase makes so much sense. And so I would actually, call for this, and I would be very happy to, after the meeting, to contact you guys. And don't feel that I bothered you. I will try to contact because I think it'll be fantastic, right? And to what it can be, the whole world will be excited to see, hey, NIH even sponsored a collaboration with China and, with people in Europe and other places to say, let's work on rare diseases in a collaboration form. Right? And this will be also for resource, and the Chinese government will be very happy to say, hey, with all the bad news, isolations, and here, one area that, could be actually work together. And maybe the government, Chinese government will be interested to give a lot more resources in this area as well. So this is something that when I see so many faces that are actually experts, and recognized names, come here, and I feel so positive. And I was really encouraged. Thank you.
好，谢谢徐校长。所以
Okay, thank you, President Xu. So first of all, thank you, President Xu, for giving us a lot of good words for this meeting. So yes, just like you mentioned, we hope to make this platform for communication even better, to really discuss. I've been having an idea that there's no boundary for science. And also I would like to respond, because yes, the meeting is always short, and maybe we don't have very profound discussion or communication. But since we have been talking about collaboration, but yes, we need to carry out our implementation willingness into the real collaboration scientific projects in the long future. We may really have some small projects or large programs among different countries. We may have collaborations among different companies, hospitals, universities, colleges. So that is the scenario I would like to see for building this platform. So Harvey landed the day before yesterday. Yesterday he gave an academic report in Fudan University, and he's been staying all day long today. So I see it's really hard for you, including the jet lag. Now you are still, could you share something with us?
Absolutely. On? Absolutely. I mean, rare diseases is something that we can collaborate on internationally. However, I am very concerned. The geopolitics, certainly originating in the United States, are not good. There's a strong move, for instance, to prevent American companies from working with Wuxi AppTech because of allegations that data that they accumulate is somehow shared with the Chinese government. I have no idea whether that's true. I hope it's not, and I really doubt it. But in the United States, biotechnology has been elevated to an area of national security. And I feel that we as scientists, as physicians, as business people involved in biotechnology, need to make it clear to the politicians of all countries that in fact there are no boundaries for doing this kind of research, that the diseases that we are talking about, although they may be enriched in certain ethnic groups, they are not confined to a single country or single geographic area. Yes, you may argue it's important to understand Chinese genetic rare diseases that are confined to Han Chinese, but there are many Chinese living in the United States, and they're going to benefit from the research that you guys do. And somehow that gets lost in this kind of geopolitical discussion of one country against another. So I think an important concept that could emerge from a meeting like this is to make it clear in some kind of statement or consensus view that rare disease research is essential, and it's essential to do on an international basis. We have many technologies in the States that can and are being shared with China. You mentioned the enormous animal resources that China has. We need access to them. And somehow emphasizing to the politicians that be all over the world that rare disease research is devoted to getting cures and understandings for disease that affect people worldwide. So, I mean, Kevin, you've taken an enormous leadership role in making rare diseases understandable, not only in China, but really many parts of the world. And I think your organization perhaps could take a lead in emphasizing the international character of this kind of research. Thank you.
谢谢 Javier。
Thank you, thank you, Harvey. Thank you for your warm applause. I think I quite agree with Harvey's presentation. The American biosafety action was just put into place. I don't like it. I know personally many American scientists. They all say that American government is converting to not a favorable one. So Harvey has been emphasizing that for all human beings regarding conquering the rare disease, there's a long way to go. We are not saying that we have done. Even in the US, there are so many diseases which could not be treated very well. So let's strengthen the global collaboration. This is the core. This is a very important opinion. But how to make China such a vast country to join the global collaboration? This is a topic we have to face. We have to face it. It's not say that it doesn't exist if we close it. It's not like that. Meanwhile, as a patient in China, as a promoter of this country, I would like to use my own strengths to promote the development of the science, to promote the global collaboration, to conquer the rare disease, to bring some changes. So I think I would like to do, because I'm a patient, so I feel urgency, and also I'm self-mobilized. I'm willing to do it. I would like to speed up because I'm a patient. I know that families could not wait. They cannot wait. So just now in my sharing, I call for all scientists, work harder, be faster, be more open, stay together, collaborate more. So because of time, so we may be not able to ask you many questions. In the next 2 more days, there will be excellent keynote speech. So I hope together you may have more close collaboration and communication with our experts during the lunch, during the banquet, or during the tea break, or anywhere, anytime. So I hope you could have a frank and sufficient communication.
Just Professor Zhang had a question with me. Would you like to say something?
这 panel 里面就是没有
We don't have people from CFDA in the panel.
Probably agree or many other agree. 这个就是要 move.
To push a therapy forward, we need to have involvement of CFDA. They should intervene as early as possible so we can avoid the bottlenecks. We have so many experienced scientists, but we need to have regulators like CFDA. We need to educate CFDA as well. Patients, family members of patients need to educate CFDA. They are learning, but they have power. They make the decisions. If they don't approve any therapy, you cannot go next step.
But if FDA said no, some policy doesn't make sense, you still cannot move to next step. So that's only my suggestion.
谢谢。
Thank you very much, Professor Jiang. It's not that we did not invite them. We invited them on many occasions. I sent a letter to them more than a month ago. CFDA is annoying. They don't show up. How to have more communication with them. So when we promote many undertakings, there are some people who turn a blind eye, turn a deaf ear to it. It's a pity. By such platforms, we'd like to invite CFDA, China National Natural Science Foundation, so that they be part of this. And we can exchange ideas, share thoughts, cooperate together. Thank you, Professor Jiang. Okay, a big hand to all the panelists. Thank you very much for your dedication. Okay, thank you for your time. We'll have